Okay, guys, so let's begin. So, how's everything, everyone?
So far.
Okay?
Are we having any surprise exam?
Yes, for sure, you're having it
We
very
need
soon.
it.
Hmm?
We need it actually.
You will get it. You will get a very tough one that then you'll say, I don't need it. But the thing is, give yourself a crash week whenever you possibly can from your schedule, everything. Because I know it's not sometimes possible for people to take time every day from your schedule and family life and workplace and stuff. Try to give yourself a few days crash where you can just sit and do a lot of extensive study. And then if you can take a break, if you want to take a break, right now you can afford to take a few days break and stuff. When you come into closer to the exam, end of November, December, you will not be able to give yourself more break time. So try to give yourself crash time. That is alternate. This is something which is coming up more like a new study style, which has seemed to do well for students rather than like our father's generation was every day study 2 hours and you're good to go. But we as our generation, we like some things instantaneous, like Netflix immediately. I want to see some. So for instant results, you can probably just sit and cram for a few days, binge studying as we call it, and then relax for binge relaxation. So a few days you can just go with family, enjoy all that. So that is another option if you guys want to use that. So the point is, get ready. Whatever you've done, get it done. Don't leave that for later because now things are going to increase more and more and more. And hopefully, easier things will be left because a lot of therapeutics is mostly done with. Still, we have few topics left, and we will do all therapeutics one by one revision again, and we will consolidate everything again. Don't worry, I'll be myself involved in all that because this week is the last week for MCQs and OSCE, and God is witness, it's crazy. So I was here till 2:30 a.m. last night, and I worked here after they left. I was here till 5:30. I took a nap for 15 minutes here, and then I continued. So I left at 8 o'clock, showered up, freshened up, came back again. So my mind is almost asleep already. And then after your class, I have MCQs till midnight again. Tomorrow we're starting early, and that's where my confusion is. The point is, we were supposed to have this guy named Route for calculations today. Unfortunately, he couldn't come from Lethbridge. So this weekend, Saturday, I'm taking here myself. I pulled epidemiology from next Sunday back to today to compensate for his absence today. Unfortunately, tomorrow would still be missed. He will not be available. We will still miss tomorrow because I'm not available tomorrow. If I had known, I would have planned differently. He's coming on the 17th and 18th, this one. So all these topics will be pulled another day. So I will remake the schedule so that it's a lot easier for you. But as of now, you need to keep in mind is calculations will be on this day. He told me that he'll be coming on this day specifically. Now we'll do epidemiology today, this one. We'll lay the concepts. We will discuss some calculations of epidemiology. Hopefully, we'll be able to still have some time. And if we're able to have some time, I'll do some of the management topics. For example, I'll be talking about where's my there you go. So I'll be talking about a little bit this one about pharmacy management here. So we'll talk a little bit about these two things, epidem and a little bit management, time permitting. Okay. So once this happens, um, we will have Islam taking next Saturday. He wanted to take one more day for the conversation about the any topics which are not consolidated in the past. And Renal, he had to go a little too fast, so he would probably be willing to spend some time with you guys in this day.
Why is Tom not coming tomorrow instead of Raul?
Yeah.
Well, tomorrow,
Discuss diabetes with
I asked him. He's not available over the weekend. That's the reason I'm compensating. Otherwise, as I said, schedule was for me to be busy with MCQs and OSCE for the last 1 week because it's more crash revision and everything, full days with them during the weekdays as well. So I wanted to be spare for them, and I wanted my team to take care of EE until I get free by 15th of mid of this month, and then I wanted to do 1.5 months with you guys. But we will still do that. Just don't worry about that part. I will still text him again today in the midday and see if he said he'll be able to discuss if he can spare tomorrow. If he does, he can come repeat diabetes and stuff, which is a good idea. And I can continue with the schedule from next Sunday onwards. So let's try that. I hope it works out with him. So that is the idea. Instead of respiratory system, he'll be doing another system here. Which one would that be? Dermatology, I guess, because he pulled respiratory with derma, right? So, and then we'll go forward with the idea. And probably here after 11th, I'll be requesting for some weekdays from you guys. And we'll discuss whichever weekdays available for everybody. Then I'll be myself there and we'll be able to fix things. Okay. Again, apologize for his absence. I was very sure he'll be able to, he's never done this before. He's taken 3 classes with me so far, and he's always come on time from Lethbridge. And he's a very, very nice guy. You will meet him and you will know.
Yes.
You met him before?
I met him.
Okay. So he will come on 17th and 18th, and we'll be good for calculations. Today we'll still do some calculations also, but they'll be epidemiological calculations. So let's lay some concepts and go forward. Okay, so what is epidemiology? Basically, we're talking about general population studies in a perspective of healthcare. You could probably have non-healthcare related population studies as well, which would probably not be under your jurisdiction, and that would probably not be defined as epidemiology. Anything, the origin of the word is from the word epidemic. You know what epidemic is? If something spreads, a disease spreads out. So if something like that happens, then that's how the term started. But now it became a big study, big branch of medicine, where we talk about not only epidemics, we talk about how the diseases happen, their background, their statistical knowledge about it, how we can try to make some modifications in our healthcare profession to prevent those from happening, stuff like that. Okay, so to begin with, lots of research method has been developed over time to make sure that we are able to achieve those goals. The goals are to prevent disease before it happens. Primary prevention. The idea is to understand how diseases take place in the first place, the breakdown of steps, how it happens. We do trials to know that first. So once you conclude how disease happens, then you say, okay, how can I try to use any therapies to treat that? Then you have stepwise medication therapy on that, self-care measures, OTC, prescription drugs. So there are trials involved in all those steps. Then after that, you say, okay, what can I do to do anything extra, which we call as health promotion, anything extra to prevent these things. So that's where stepwise trials are there too. So there are going to be different domains within epidemiology. There are different domains. Domains means categories, which are going to be talking about therapy. Some are going to talk about diagnosis. So there are different domains in there. At least 4 are which we're going to touch in our topic today. And I will explain as we go which domains we're touching. Then, how are you going to achieve all these goals in epidemiology? You would like to know some stepwise methods for yourself as a healthcare professional. You may not be doing an out there research in a research industry or something. How does it apply to you today as a pharmacist sitting in a community pharmacy, for example? You need to know how to ask good questions, either to yourself or to your colleagues or to your healthcare professionals like doctors and stuff, and above all, to your patients, right? If you know how to ask good questions, if you're able to break down some stepwise process in asking good questions to acquire good information, then you'll be able to achieve those goals. At least it'll be a good starting point to achieve the epidemiological goals. Let me make it simple. You need to know few steps, which we'll do today. And if you're able to put them in your daily practice or routine as a pharmacist, you'll be automatically able to come to good conclusions because that lets you do good questioning. We have some mnemonics we will remember and we'll go forward. But before that, we'll try to know a little bit about what clinical trials and all the idea goes about. We already know what clinical trials are, right? You do research studies to compare some drugs sometimes, some new medications come in the market, etc. Even though you have a lot of trials when it comes to diagnosis and other fields of medicine as well, non-pharmacy fields, we're not talking about those today. We'll be talking about new treatments or existing treatments. By the way, sorry, there might be some updates in the epidemiological lecture of today. You basic structure is the same of the lecture which you have. Two possibilities: either you make notes on top of whatever you have on the sheets, or if anybody wants new set, I can always provide that, no problem. Okay, there wouldn't be too many differences, just a little bit differences. So we'll be talking about new treatments and we'll be talking about some existing treatments. And you don't have to memorize this all. This is just for concept building. Okay. Clinical trials are experiments or observations done in clinical research. Experiments and observations. Two things we've spoken about. And as we're going to go forward, when we divide the types of trials, we will define the way and we'll say there are some observational trials and there are some experimental trials. So that's what we're talking about. Such prospective biomedical or behavioral research studies on human participants are designed to answer specific questions about biomedical or behavioral interventions. Don't worry, it's just an idea to say that you're going to be exploring some different fields or domains, as we said. I want you to pick some information from the next 3 or 4 slides, which I'm going to show, and make some notes on those. What is that? From this slide, we're taking information about the durations of trials phases. So what happens is your clinical trials take place in different phases. Different stages are there for each clinical trial. A drug, let's take drugs as an example for clinical trials. When a drug is, the idea is initiated or incepted, it is in a preclinical stage. That means in the lab where they said, okay, probably if this is the necessity, we need something which for probably breaks the cell of bacteria and da 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 da. So they calculated something on theoretical basis. Then they tried to devise a molecule in theoretically first. Then practically they try to see if it is possible pharmaceutically. Then they make a molecule. This is preclinical. This can take you several years, long time. They're doing research, lab works, millions of trials and errors. They will try and fail again. They will try again, fail again. They'll keep trying to improve on a molecule as much as they can. Maybe they'll come to a point and after 10 years and say, doesn't work. Maybe this formula is not going to. Let's start from the beginning. So they start something else. So that's why millions, billions of dollars are invested and tons of time is invested in preclinical stages. So the things which you're taking from this slide for me, you're going to note down for yourself in your notes. First thing, preclinical takes several years. So we're going to talk about 5 phases. First phase is preclinical. It's going to take several years. Then, actually, what you can do is you can make columns because we're going to put more information on each phase as well as we go in the coming slides. Okay. So first thing we said, preclinical takes several years. Then, after it's coming out of the lab, now you say, okay, I think this molecule is good to go. We have created a medication, but we're not sure if it's going to be good practically speaking. We've tried it on animals. We've tried it, all that stuff. Looks good. Safety profile is good. It has helped to treat the disease of the animal, for example. Now let's put it on humans. So phase 1 is where you start with the humans. Phase 1 is when you start with human testing.
And what about with the animals?
Animal was in the preclinical.
Still.
Yes, in preclinical, everything was doing, and animal testing was done here as well. Okay, then we just started testing on humans from phase 1 onwards. So, phase 1 is what? In phase 1, you're just trying to have some healthy individuals. They're not diseased, they don't have the disease which you're trying to treat. So, you're trying to find out in a normal human population, healthy people, is this medication tolerable, acceptable? Before we give to the diseased guy and he dies, let's try on healthy people first. Plus, you want to see, first thing, let's say they're healthy people. So then you want to see a range of the dosage. For example, in animal studies, we usually give them sometimes 4,000 to 6,000 times the dose as well, just to see. And usually animal dies, but it's just to see the limit until till which limit was bearable, just to see the toxicity levels of the drug. So in animal studies, the range of the drug dosage was really big. In phase 1, the range is relatively smaller, but it's still not final, the dose. For example, if maybe statin was in a study here, they might be saying starting from 2 milligrams all the way to 200 milligrams, maybe. Whereas here, maybe they were saying from 2 mg, I want to give all the way till 1000 milligrams on animal studies.
We can say this is in animals or the preclinical trials to find the minimum effective concentration and the maximum effective concentration.
In a way, you can say that you're trying to find the minimum to maximum range, above which would be toxicity, below which would be subtherapeutic. So you found that dose. After that, what you can do here in phase 1 on humans, you're going to have much lesser range, shorter range. You're trying to specify a little less, and then you're trying to see, was it okay on his kidney function? Was it okay on his liver function? Is he able to tolerate? Is he having any side effects, etc.? So you're trying to check all these things in phase 1, and we're going to put all the other points in the coming slides as well. Safety and all that, we'll do that later. This is just to give you a brief idea of how things are going forward. Then you come to phase 2, and the phase 1 took you just few months, not too long. It's just healthy people. You're just trying to see if the medication is okay.
This people is volunteers, right?
Usually volunteers only.
Yeah, yeah. We can't go for people in the street. They come and.
That's right. Yeah.
That's right.
Yeah. You can't just be a guinea pig without knowing.
So usually they pay them. Yes.
That's right. Few months ago, actually, I was looking at an advertisement online in Canada that will pay you $999 per week if you will be willing to join a new trial. So what they'll be testing you. They'll be in phase 1 trial. And what if after a few days you become a rabbit? You don't know. What will you do with the money? So this is phase 1. Then phase 2 is, now we're talking about diseased patients. These people have the disease now. And the group is bigger as well. The numbers we'll discuss in few minutes. Here the group was smaller. Now we're talking about few hundreds of people. Months to years it might take. Right? It could be just 1.5 years, 1 year, 6 months, or it might even be 4 years. Rarely phase 2 will go beyond 4 or 5 years. What's happening in phase 2? You're specifying dose even further. You'll come to a final conclusion of the dose. This much milligrams is my optimized dose. That is the final dose which will go for the rest of the life of the drug. You chose the final amount here. So if somebody was to ask you, when do you start thinking of dosage? is phase 1. When do you finalize the dose? Phase 2. If they don't ask you about start or stop, if they simply say which phase decides the dose, phase 2 decides the dose. Okay? Phase 2 is when you decide the dose, the final dose. So now you've tested the drug on the diseased patients as well, on a moderate group size, and you've tried to calculate whatever you could. You finalize the disease, everything, sorry, the dose and everything. Now comes a point where you say, okay, now I have a preliminary, or let's say rough draft ready. Rough draft means just like initial document of the drug. Now you've created a, the monograph of the drug which you get with the patient information, leaflet, package insert, with the medication in the box. That is the final product, right? But the initial draft was there at some point in time when they had decided the dose, when they had decided the indications of the medication. So that initial draft is ready. Now they will give it to, now they will apply to FDA different places wherever they necessarily have to, as we will see in the coming slides. They apply to FDA and they say, here it's Health Canada, by the way. So they applied to Health Canada and then they say, this is the drug. We tried phase 1, phase 2. Animal studies were done. Healthy humans and diseased humans were done. We concluded these are the indications for this medication. These are the side effects for this drug. These are some interactions we found out. This is the dose we want to try. Stuff like that. Once they get the approval of this first stage, say okay, looks fine, but do more testing on the long term. So now they're going to come to the phase 3, which is on large population, and it takes years to decades. That means it can be few years, it can be 5, it can be 10, it can be 20, more than 20, depends. So this long-term study is done again on diseased patients. Here you're establishing long-term efficacy, long-term side effect profile, because some side effect might not happen today, it might happen after 10 years. So that is something which you're trying to establish here, right? So lots of things are happening in phase 3. It's the biggest and the largest of the, so far phase 1 was small, phase 2 was longer, phase 3 would be even longer, and phase 4 would be ongoing forever. So that's how small to larger the duration increases as the time goes by. But preclinical was before, don't include that, that was way longer. So before phase 1, these are how the phases would be, the duration wise, I mean. Okay, now after this you try everything, you find out whatever you could, and then you go to FDA and say, okay, my drug has proven to be a success in phase 3 trial. It is treating the disease, it is having limited side effect profile, interactions, whatever, all the thing. This is the final approval from FDA, or in Canada it will be Health Canada. Final approval happens, you get NOC. NOC means no objection certificate, and your drug gets a new DIN number. What is DIN number? Drug identification number. So each medication will have a small little specific number on the medication in Canada to identify the drug. So that DIN number is issued for you. Now you have DIN, now you have NOC, now you can launch in the market. You can do proper marketing, brand name, you can launch it in the market. And once it's in the market, then it starts something called the phase 4, which is an ongoing phase, never ending, not stopping. That means all of us are taking whatever medications today, you're in phase 4 right now, because this is post marketing surveillance. So at any point in time, a drug is showing to be dangerous for the population of any, we can pull it back. We can say, okay, despite we went through all the rigorous process, despite everything was approved, now since it's in the real market, since past 15, 20, 30 years, we found that thalidomide can cause you phocomelia, or you can find out that cimetidine is causing gynecomastia. These are long-term side effects. Then you pull out the drugs from the market. So essentially, this is still part of trials, still guinea pigs. But the idea is until you're able to come to a conclusion that this drug is really good for long term, a drug is still vulnerable to be pulled out. So that is on the entire world population. So that's unlimited population. That's ongoing, infinite time. Okay. This is just the brief.
If it's like something happens after phase 4, can you still sue the manufacturers or
Mhm,
no?
mhm. You can definitely. You have the right to sue a manufacturer if a drug was not supposed to do something and it did, and if you're able to prove that it did, that's where the problem lies. A lot of such trials, not this trial, the case trials, the court trials, the cases, they fail because um, you're not able to prove that this drug caused this for me. The causation. The problem is biggest of these statisticians out there till date are not able to conclude that smoking causes lung cancer. Why? Because they can say it's association, it's not causation. They're two different things, right? For example, if I go to Tim Hortons at 9 a.m. in the morning, I find a person there at 9 a.m. I went next day at 9 a.m., he was there at 9 a.m. Third day, I went there 9 a.m., he was there 9 a.m. We cannot say that whenever Mohsin goes, that guy comes. This is not causation. My presence didn't cause him to come, but it's just an association. At a time, both of them are there together. So this is something which is an association. It's not causation necessarily. We cannot say with proof. What if he's just looking from his window, waits for me to go, and then he follows? It is possible. It could be causation, but we cannot prove it. That's the problem. Smoking, can it cause lung cancer? We're not sure, but it is associated with lung cancer. Those people who are smokers have a higher risk of lung cancer, yes, but it's still not causation. So this is the problem. You will not be able to prove it to the court that this medication caused me this problem. They will say you are vulnerable, you have genetic problem, or you are having kidney issue, or you had this. They will find something or the other to find a defense for
Yeah,
the
but
drug.
my question was like FDA is such a huge body, like it's the everything. So if it, the court or the manufacturer can say if the FDA is approving my drug and if it caused something wrong with you, like if my question was, can a person still sue the manufacturer because even the FDA was like
You
sure
can.
get this drug.
Yes, definitely. FDA wouldn't be responsible for that because any documentation, any approval, either the rough draft or the full draft, whenever all these approvals are done, it is very clear in their terms and conditions everywhere. Out of those 1 million pages document, the contracts they have, per drug has like hundreds of files which get approved, by the way. Their pharmacokinetics, their dynamics, their bioequivalence studies, tons of things. Bioequivalence itself will have 15, 20 different files. So the thing is, once all of that get approved, they have in their contract, FDA says, we're not responsible, okay? If you're bringing a drug in the market, we're just saying that by your trials, it looks okay, but if tomorrow somebody dies, don't come to us. I mean, you're responsible for your drug. So that's why they're responsible. You will not sue FDA, you can sue come manufacturer for sure. And there are lots of lawyers out there who are specialized in pharmaceutical suing. And sometimes they say, if you use this medication for 5 years, and if you feel that you have kidney stones, then you can contact me. And this is my number. They're advertisements, right? And the reason why they do that is because they are specialized and they found some tricks to get some money from the pharma company. They will not win the case, and pharma company knows, but it's just that it defames them. It defames their product. It demarkets it. That if I was to sue a company and I say statin caused me lung cancer, for example, and then I can just say, even though I know I'm not gonna get the win the case, but it's in the public, it's in the news. I'm putting Facebook ads, I'm putting newspaper ads, whatever, and then the company will come on the table and say, okay, what do you want? To get your mouth shut. So that's where the lawyer will help you facilitate some money, some for him, some for you. So this is where the game lies. Okay, now You will never win, by the way.
So, do they need to finish the whole stage, or they might be in stage two? They said, "Oh, it's not gonna work. Let's start again."
Yes, at any point, stage preclinical, one or two or three or even four, a drug life story can stop. At any point, the drug misbehaves, or any point the drug shows to be bad for human, pulled out. Billions of dollars down the drain. But a lot of things have happened, to be honest with you. If any point in time, whenever we were able to lose a drug at a bigger stage, at a later stage, we knew that it came this far. That means we are close. Even if this drug fails, no problem. They don't have to spend again billions to make new drug. They are able to try to modify same molecule in whatever ways. Cimetidine being an example, they knew ranitidine, famotidine, other sister drugs can still be good. Cimetidine is bad, pull out of the market where other drugs can be used. So they're able to spend less money and still find some other sister drug.
Is that possible to let any drug to go out of the market? Uh, like 10% is gonna work and 90% is not gonna work, but this they said okay, like for example cancer, if they found a drug that will work only 10%, can they let it go? Like maybe they would say okay, he's going to die anyway, so at least 10% might he live for another 10
Good.
years.
So there are lots of places where compromise lies. There is a trade-off at several places. One of the examples I will give you is alendronate. Alendronate coming to market, bioavailability studies were failures. Until death they are in real humans too. The bioavailability is terrible. Less than 1% of the dose is absorbed. That is, if you give 100 milligram to someone, less than 1 milligram will be absorbed. It's that much of a difference. And it made through to the market. Why? Because whatever gets absorbed still does its job, right? So whatever percentage of whether it works, some percent, not full, to answer your question, yes, definitely that can happen. And it totally depends on what it's bringing to the table, what it's bringing to the market, what is going to offer value to the patients. So if it's worth it, then definitely yes.
What about the side effects? What about if they say okay, we found the drug, okay, it's working very well, but a lot of side effects?
No, so side effect has a limitation there too. Benefit versus risk. So they will say, okay, in order to help a patient for flu, we're ending up giving him cancer. No, this is not a good trade-off. Versus if we're trying to treat someone's cancer, and in order to do that, if you end up giving him flu, no problem, we can do it. So it's more risk versus benefit. Which side is more heavier out of the two? It's usually based on lots of criteria which they have. Tremendous criteria is there. How many side effects? If it's having kidney, a drug, for example, if it's going to damage kidney as well as liver, that is automatically a red flag. A drug is automatically put for second appeal. That means first application failed. Because the way we think in pharmacokinetics is if a drug is renally excreted, okay, person's liver is going to help metabolize. And we have alternate pathways. We have an outlet. We can figure things out. We can attach some alcoholic groups or do whatever to the molecule and make it a little more hepatically. So make a drug balance. Versus if something is hepatically metabolized and it's damaging liver, we'll say, okay, no problem. We still have kidney. We can do something through renal route. Da da da. But if a drug is doing both, it's automatically a failure. So I'm just trying to say, give you an example that there are lots of criteria which they have to fulfill for the side effect profile. But one of the primary things, even before efficacy, is safety. In the trials, one of the first things they want to approve for is safety of the drug. Then we'll think of efficacy and all those things later.
So, like my question about the AIDS.
AIDS?
Most of them they die 3 years later with the liver because of the drug.
Right.
So is that the same case?
Exactly. So if someone we're able to eradicate HIV for them, a lot of antiretrovirals, you're saying they have a lot of hepatotoxicity. Totally agreed. A lot of liver damage happens over time. But again, the bigger purpose is we're trying to save person's life. We're trying to achieve HIV remission, and we're able to achieve that. No problem. If hepatotoxicity happens, it happens. Okay. So this is just the general idea, and let's move forward and add some more on all these four phases. Now we're talking about number of participants. So let's try to memorize some numbers now. Okay. Preclinical is on just on animals. Oh, you're going, Imad? Okay. Is
Yes,
that what he
boss.
said? Okay. All the best. The lecture is recording. You can watch it later. Okay. Okay. So preclinical is on animals. Lots of animals. You don't need to know the number of animals wasted in the process. That is
We could
irrelevant.
start from the people who's against that.
Then phase one, 20 to 100 approximately is what you start on the phase one. Remember we said limited number of people.
Yes.
So were they diseased or were they healthy?
Healthy.
Phase one, they were healthy,
Healthy.
right? So healthy people, approximately 20 to 100. Some places you will find the number 80, 20 to 80. That is also okay. Actually, more resources I found with 80 than 100. But this is also just a rough idea, 20 to 100. Then phase two, the basic idea in phase two, the number is less than 1,000. It can be 100, it can be 200, it can be 500, it can be 800. Basic idea is less than 1,000. Phase two. Are they healthy or diseased?
Disease.
They are diseased people. Limited number of people we're doing our testings. Then we applied for the rough draft, initial draft to FDA. We said, can we go for phase three, please? Okay, what is your dose? This is my final dose. The idea is if before this you go to them, phase one or phase two, they will say, huh, you don't even know your dose. First go decide your dose and then come to me. So that's when phase one, phase two, you're deciding your doses and starting to making your rough draft. Done that, approved for first draft. On diseased patients, you do bigger population, 1,000 till 3,000 approximately. Large number of people are done here. In this large number of people, you do all your trials, good to go. Then you get something called the FDA review. You apply to FDA for the final approval to confirm the safety and effectiveness one final last time, and then they approve. Then you go to phase four. Yes, please.
And in the phase 3, are they all disease patients?
Yes.
And still volunteers? Still volunteer
No,
patients?
these are
Or
diseased. Actually, here's what happens. The recruiting of the patient in phase three would be different than the recruiting of patients before phase three. Before phase three is all volunteer. In phase three, it could be volunteer or non-volunteer as well. For example, there might be a situation where a patient is not able to make a decision, mentally incapacitated, cognitively impaired, unconscious person without a decision maker or guardian around him. The different possibilities might be there.
Or hopeless guy.
Cancer, maybe last stage of cancer, and you're deciding, okay, calculating, I think he won't survive more than 2 weeks. Then you do what? Then you can propose if the person is able to decide or family member is there, they want to try this drug, this is volunteer. But they say no, if then you can't. Versus a person who cannot answer for himself, there is no guardian. A lot of times this happens, especially in this part of the world where we live. A lot of family ties would be probably different. You may not have a family member around you when you have cancer lying in the bed since the last 5 years. And if you're on the deathbed in a hospital, last 2 weeks given to you from the doctor, and you're unconscious, then they'll make a decision. You know what? This guy's dying. And you know what? We have a new drug in the market. We can try it. This is your subject now. He's not a human anymore.
Is
You
that
can,
possible to
you,
sue him later?
why not?
When he
But
wake up?
but the
If
thing
they
is,
found out.
yeah.
Yeah.
But the thing is, you might have a rationale to defend yourself. At any point in time in medicine, you have a rationale to defend yourself, game over. You have the rationale that, okay, my rationale was he was done with, and there was no one to give a consent. We tried our best. We gave the best shot we possibly could, a new drug. We thought this is better. Researchers say this. Manufacturer says this. Your defense. So you can be safe in the court. Now, the thing is, that can definitely happen, but there will be volunteer plus non voluntary as well. Self recruits are here too. Whereas you cannot do that in phase two. You cannot just randomly give any drug to anybody unless this is a volunteer, unless somebody agrees to do that. Whereas in phase two, what happens is, for example, one of my cousins, he passed away a few weeks back. He had bladder cancer in UK. One of the new monoclonal antibody they offered him that, do you want to try this? He said, I don't know, Mohsin, what do you think? I was like, well, it's a new drug, might as well try. His wife said no because she did some research. And you know, when someone who doesn't have medicine understanding, and when they look up the side effect profile, it's like starting from hair loss, going all the way down eye, nose, mouth, ear. Everything is going to go wrong till my toe.
Google.
Everything. This is what Google will give you. So she saw all the side effects. She said, all this. I said, Bhabhi, but then all this side effect, he's having cancer with bladder. He's going to die anyways, right? What's the problem? So, but the thing is their decision eventually. They didn't decide to go with the drug. They accepted it. You know, when it becomes the religious thing, it becomes like, okay, I've accepted my fate. I know what's going to happen. It's okay. So then there's nothing you can do to convince them. So he fell out of the volunteership. He did not sign the contract. The point is, in phase two, the drug was. So in phase two volunteership, they will offer you. They cannot force you. Even if you're unconscious, they cannot, because it's not gone to even the first draft. FDA has not acknowledged it as an investigational drug yet at all. By the way, since I'm on the topic, if at any point you find the word investigational drug, usually they're referring to phase three. Okay. Phase three is when the drug gets some kind of respectable name. Until then, it's more like a RD467895. This is the name of a drug, which you cannot recognize. So until phase two is that, and phase three comes, they say NZT 30. Oh, there you go. That's a drug. So now there is a drug of name of a drug. So it becomes a little bit pronounceable by general population. It's still not called statin. It's still not called any particular name, but at least it has something. Then only after FDA approval and you get the DIN, that's when you apply for a market name. I want generic name to be called as trastuzumab. I don't know where they come up with these different ideas, but they somehow come in with those ideas. Trust me, if you see the chemotherapeutic agents list, the new types of monoclonal antibodies are there. I was sitting with MCQ students last week that how can we memorize them? It's impossible. It's impossible. So unless you're a even biological experts, those who know all the biologicals, it's not possible. So then comes phase four, and then you have people more than 1,000, as we said, rest of the world is involved. Let's do this again. Right now, what we're pulling from this chart is the number of participants. We said approximately 220 to 100. The criteria is less than 100 here. It could be 50, it could be 80, it could be 100. Here, the criteria changes for phase two. Less than 1,000 is the idea. 100, 200, 500, 600, 800, whatever. Phase three, usually the criteria is less than 3,000. Okay. And then we go in phase four, a lot of people, everybody. Okay. Let's pull more information now that each phase, what is our goal? What we're trying to pull out from each goal, each phase. We already said in phase one, you test it on healthy individuals. We know that. So the info you want to add to your list now is number one, safety. Number two, dosage. Number three, side effects. So three things we have said so far are what you're doing in phase one. By the way, this is not preclinical. This is the phase that started officially. Okay. Phase 1 is tested on whom? Sorry, preclinical is tested on whom?
Animals.
Animals. Then phase 2 tests on larger group of affected individuals. When you say affected individual, we're talking about diseased people. Okay. What are we testing here? Again, for efficacy and side effects. Now, the idea how you can differentiate between phase 1 and phase 2, which they might ask on efficacy. What are you testing? What are you checking for in phase 2? Your main priority answer would be efficacy. If they give you all the options, efficacy, side effects, both of them are there, you will choose efficacy. Why? Because in phase 1, you cannot test efficacy, there is no disease. There is no disease. What can you test it on?
Dosage and side effect. We can.
That's right. So there you test it having safety, dosage, side effects. Number one priority would be safety in phase 1. Whereas here the priority is efficacy. Okay, makes sense. Now you have the diseased patient. You have a disease to check the efficacy against. Now phase 3. After there's one more rough draft. I keep emphasizing this word here because they they've asked in the past something similar. So between phase 2 and 3, there's a preliminary draft which we call as the, there's a name for it also. Proof of concept. Proof of concept. That is the rough draft which I've been using the word for. It's called proof of concept. That means, do you have some idea with you what the dose of your medication, etc.? Gone through that. Now test a new wider demographic population. What is the patient population in phase 3?
Less than
Less than
3,000.
3,000.
Yep. So prox 1,000 to 3,000 is approximately. Test for long-term effectiveness because it's a long-term study. What is the duration we said of phase 3?
Years.
5 years.
Years. Months was phase 2.
Decades.
Phase 1 we said was just months. Phase 2 we said would be months
Or
to
years to
years,
decades.
whereas phase 3 would be years to decades. So we're talking, just to give you a rough idea, no need to memorize or write this down. Months could mean probably 6 months all the way to 18 months. Phase 2 approximately you can say 1 year, or maybe even 6 months all the way to maximum I've known about is 4.5 years. Phase 3 could be 1 year or so, or 2 years onwards all the way till more than 30. There have been drugs for that long as well in the market. By the time they come to the market needs have been changed. Then, and comparisons with other medications happen here too. So there are two things you got to take home from phase 3. First, long-term effectiveness. Number 2, comparisons with other medications. That's where RCT starts. Randomized controlled trials. That's where we start them. That if this statin is doing this, what about fibrates? What about other types of statins, etc.? So you're trying to compare them against other drugs. Now keep in mind, phase 3 trials are sponsored by the pharma company. It's your baby product, you want to work on it, invest money and get it more promoted. So you do trials investment. Versus phase 4 RCTs will be in the open market. Everybody else can do RCTs as well. That okay, he says his drug is better than mine, let me prove it wrong. So then they're going to start trials on the other drugs too. So it's open market challenge in phase 4. But in phase 3, only pharma company, you yourself can do it. Nobody else cares until it comes to the market, right? Then FDA approval happens, treatment determined effective and safe for public. Then you can continue efficacy and effectiveness and safety at any point. Today you're using a medication, you find a new adverse effect, you have the authority and the right and the duty to report it back, right? So this is what we're talking about here. Phase 4, you can always report back. This is the keyword, can be taken off the market if necessary. Okay. So I'm going to pause the recording. Did I start? Yes, it started. Okay. So dose of a medication is defined in which phase?
Phase two.
Phase 2. Yes.
Optimized dosage.
Phase one. Phase one.
Phase 2.
Phase one.
Phase 2.
Dosage finalizing. Final
Right.
is in 2 and 1
So
is stuff.
phase 1 is when it starts, phase 2 when it's finalized. Well, if it's as this way, it should be 2, where the medication is defined. But then the idea is basically to give you 1 is to start, 2 is to finalize. Okay. NOC from Health Canada is provided after completion of which phase?
Phase 3.
After phase 3,
After phase
you get the NOC?
Yes.
Good. DIN is issued after completion of which phase?
Phase 3.
3?
Yes.
Yeah.
3.
When the FDA approval happens, same thing.
Oh yeah, okay. After.
Yeah. Which phase is also known as post-marketing surveillance?
4.
Phase 4. Competitors comparison takes place in which phase?
Phase 3.
Three.
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 Comparison.
3.
Now you are confusing the word comparison with this comparison.
It's 4.
This is comparison of competitors. Comparison.
Oh,
Those
yeah,
are
yeah,
comparison
other
against other drugs in the trial versions. The word competitor is the idea. In phase 3, the drug is not in the market.
But
There is
the
no
company-
competitor.
But the company comparison with other drugs.
Yes.
Yeah.
Normal medications, statin versus another statin versus fibrate, drugs versus drugs. You're doing your own comparisons in-house.
Mhm.
The competitor
Oh,
means
yeah, other
in the
drugs.
real open market, when the real drug is out in the market, when your competitor is going to be challenging his drug versus your drug, brand name versus brand name, all that picture comes only after the drug is in the market. Phase 4. That's the intention of putting the word competitor here. But the normal drug comparisons, general trials against drug versus drug takes place in 3. Okay, good. Are we good
Sorry,
with the trials? Yes,
uh,
Ibrahim, go ahead.
which time they opened the world market? For example, Plavix, the company had to take it for 10 years, or adrenaline, or for 5 years. It's in there because they will make more profit. Which phases in this 2 or 3? I don't know if I could give you the idea.
No, sorry, brother, I didn't get your question.
So, for example, I have a new medicine like Plavix,
Okay.
and I had to make a profit exclusive marketing. It's belonged to my company for 10 years or 5 years.
Oh, okay. So you're talking about the patent.
Yeah, patent, and then they open. So in phase 3, it's, for example, for the Pfizer, it will be stay, and then I will give the molecule for the other companies or the world.
That's phase 4, buddy. So what happens is, after the drug is out in the market on phase 4, when FDA approval happens, they get some legal documents signed elsewhere, so many places with the government bodies, which we call as patent, or you can say patency. Patency rights. And yeah, thank you for bringing my attention to that, Ibrahim. That is an important point I missed out.
No worries. Thank you.
So patent means that now you're saying I spend millions and billions. Now if I come to market, there's a thing called stealth marketing. Other companies are sitting there and waiting for you to come with a product, and they will like, okay, you do the hard work, I'll reap the benefits. So then you bring a drug, I'll copy it, make a generic, and I'll sell in the market. They have patency on that. They say, okay, you know what? I have patency. If I release this drug, I want at least 20 years signed by myself. I cannot have anybody else take benefit from my medication for next 20 years. Only I will sell it. I'll take the money. I will recover my cost, make billions on top of it, and then I will let it out in the generic market. By average, usually, unless specified otherwise, the duration is 20 years in Canada. Patent law, 20-year by brand. Brand market says, I made a drug, Abilify. The generic name is aripiprazole, and tomorrow nobody can make a medication like me. And the patency, there's 20 years patency finished just 4 or 5 months back in 2018. And now we have a generic of aripiprazole. Nice little bottle, much cheaper, and a lot of people can afford it. But the problem is, it's not like tremendously cheaper. Like the thing is, if I'm able to pay $168, I wouldn't mind paying 154. I mean, like, if I can pay 154, I won't mind paying 168. $14 wouldn't make me poor and make you rich, you know. So the thing is, there's very little difference now. But long story short, other markets can make after that patency law is over. That patency can be any duration. 20 is just a number. 20 is just an average. If a drug manufacturer says, I will go with a patency of 20 years, okay, Health Canada might say, wait a minute, or FDA might say, wait a minute. Uh, I don't think you spent too much on this drug, because this drug you just did trial on 3 months here, half a month here, 1 year there, you're good to go. So you spend only much lesser than a normal patency law. So probably 5 years is good enough. Maybe they negotiate on some terms and conditions. They put their financial problems in front of them, expenses they spent on lab and this and that. Then they say, okay, I think in 5 years you will be able to recover your money, and in next 5 years you'll be able to make profit. Okay, 10 years. Agreed? Agreed. They sign a contract. So it can be modified. The duration can be less or more than 20 also. It can be 30 years also. Oh, it was an HIV drug. We spent like way more than it should have been. Okay, we'll let you 30 years to make your money. Then others can do it. Now, trick comes in the game. The problem is, patency laws are country bound only. Each country will have different patencies. So if I'm launching a drug, I will probably say, okay, it's my new drug. I will have patency in Canada, patency in USA, separate laws. In every government body, you will have different patency law going. So you have representation. Maybe I'm Pfizer, for example. I have an office in Switzerland. They're having patency law applied in their country. I have representation in an African country. They're having with their own government there. All of this is patency is done. Then some country where you missed out or you didn't do, then you have, for example, a company like Cipla in India. What would they say? You know what? No patency here. Sure. Then they will look at the molecule, they will duplicate it, make a new medication generic, and do it.
They can do it in another country has a patency, but sell it in India.
Yes.
Oh yeah.
Now here's the problem. Even if there is patency in a country, and if the rules and regulations of government implementation is weak, because making a rule is different than implementing a rule, right? So if making a rule is easy, patency in Pakistan, good. On paper it is there, put in the drawer and lock, and then they say, okay, now how much money you're going to offer me to let you make the drug? And you still make the drug, and their rationale is, we're helping poor people. Like, like they're poor. We got to give them the drug. And 20 years you want them to die? No, but they're making billions themselves from somebody else's. This is stealth marketing. But anyway, this is just the idea of patency.
Is that possible? A company to make a medication and they do their best. Like for example, for the diabetes, they found a drug that will really 100% treatment, but they found out there's a 50% chance to cause renal dysfunction. Is that possible? The FDA or healthcare, they approve it, okay, and it's a choice of the patient to take it or not?
Yes, risk versus benefit again. Same question which we answered before. If it feels like benefit is more than the side effect, whatever is heavier, we will go forward. We will leave it in the market as an option. It is still possible.
So they can approve it as an option, and it's up to you to take it or not.
Yes. Unless it's a very, very sure causation, For example, pioglitazone causes you bladder cancer. Its causation, it is proved by some trials, at least 3 trials. So if that is proven, pioglitazone is slowly being withdrawn from the market. Firstly, what happens? A lot of people will stop purchasing it. A lot of prescribers will stop prescribing it. Automatically, manufacturer says, fine, he takes it out of the market because it's no benefit. He's spending money, effort, energy on the marketing and all that. When there is no return of money coming back, he stops. So to kill a drug, it happens automatically, not usually forcefully. It's the gradual decline when people stop prescribing because side effect is in the market. Everybody knows pioglitazone is going to cause bladder cancer. So they stop prescribing, goes out of the market. Rosiglitazone instead you started using is more instead of pioglitazone, stuff like that. So this is where you don't have to take an action, it automatically happens.
Is puberty still there or is it withdrawn?
It's still there, much lesser used than before. Okay, so clinical studies, we can divide them into descriptive or analytical. Descriptive means I can see something happening and I will just say, okay, that is happening. I didn't do anything. I didn't try anything to change that happening, that event. I didn't intervene. I didn't make any change. I just described it. I just called blue as blue. I just called red as red. That's it. Just describe whatever is happening. There are at least 3 different types of studies. There can be more as well, but for your scope, case report, case series, and something called as meta-analysis. These are what? These are just descriptions. We see something, we describe it as it is. We discussed what is case report. It can be a single report coming from any part of the world. That I found a patient, for example, in 1884. First report, case report came on in African country that we found a person having a simple type of a virus, which seems to bring person's immunity down. Sorry, this is 1984. My bad. I went a century old. So 1984 in African country, they said, we found a virus in the person's body, which we believe could be a virus. It looks a different type of virus. It was a retrovirus. It wasn't a general virus. We're not sure what the entity is, but we found that it brought the person's immunity down and looks like it is contagious. Just a case report from African country. Then 1986, by 1986, they had collected above 30 such case reports from different parts of the world. More than half of them are from African countries. So someone sat down and made a case series out of it. Took all the reports, compiled them together, made a case series. Different case reports together make a case series. That out of 30 patients, we found this demographic, this, all stories put together in single report. Again, you did not do any kind of intervention. You didn't do anything to change it. You just described it the way you found it. Description. What is meta-analysis? Meta-analysis is actually one of the most highest levels of evidence. There will be case reports, case series, these two. Then there'll be several other things which we're going to see. And then at the top will come meta-analysis. There'll be case control, cohort, all those things are in the middle. We're going to talk about it in the coming slides. But meta-analysis is right at the top. That means, what do you do in meta-analysis? You pick up different RCTs, not case reports. Case reports are just reports. But you pick the proper RCTs, randomized controlled trials. Or you pick up different case series, cohort studies, different types of good level studies, and take only their statistical information, put it in a single format, present it. You didn't give your opinion there that I think this study is superior to, no, nothing. No opinion of mine, no interpretation involved, just description. As the studies were compiled together, presented, description only. This is what meta-analysis. Analysis of different types of, but again, the word analysis is misleading because it is not an analytical study. It is not an analytical study. It is just a description. You pick up statistics, present them as stats. The only thing which you did there, though, was different studies are done on different study designs. Sometimes they're not comparable. Sometimes you're not able to compare apples with oranges. They're different. First, we do is we bring them on same level. Firstly, we try to pick only all the apples from the market, try to compare them. Then pick up all the oranges from the market, try to compare them. Then we bring them on the same format, try to make all of them apples or make all of them oranges. Try to bring them the same format, comparable format. And then we say, okay, statistically, these studies say this much causes dyslipidemia, these are da 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 da. You've managed a bigger statistical number. Study from Africa, study from North America, study from South America, etc., brought together. That is what meta-analysis, description only, okay? On the other hand, what is an analytical study? Analytical study means I go beyond description also. Now, I'm not only describing as it is, I'm going to give some analysis. I'm going to give some interpretation that what I think about the study. I think this study should have been da da da, or this study design is not good, or this study concluded this way, it should have concluded that way, or maybe the result of these studies are showing we need to do this in the market, etc. What is this? Analysis. Analytical studies could be either observational or experimental. Observational means you're just observing without interfering. You're just saying, okay, if some people did smoking, others did not. They developed cancers, others did not. This is my observation. Now, if I say, you know what, if I was to do some intervention and experiment on them, I will try to, after a while, tell them, you know what, guys, you stop smoking, and you guys, you start smoking. So maybe I'll do something, flip over, da da da. This is experiment. Even though you will not tell them to smoke, just an example. Okay? So that's what experimental versus I was just sitting and observing them and I was describing it and I was doing more than description. What is that? Analysis. Now I'm analyzing it as well. That is analytical observational. For example, cross-sectional study, cohort study, case control study. Versus if it was an experimental study, we'll be talking about something like RCT, randomized control trial. It's a proper trial. You were able to do some kind of intervention. That those guys who were taking medications, others were taking placebo or not, then you can do crossover if you want. You're changing something, you're experimenting on them. They're guinea pigs for you.
So the experimental just you, and just for example, like to ask them to switch, and we'll see what's gonna happen.
Yeah. Okay.
There's
Just
a
like
question.
Hmm.
It's like, who are these people doing these studies? Like, are they pharmacists or?
They can be pharmacists as well. They can be pharmacists as well. Totally depends on whatever your backgrounds are from. As I said earlier, you're in the field of science as a pharmacist yourself. If you feel that you know there's a problem which needs to be solved in your practice, if you find something on Cathets website, anywhere you found some open-ended problem which needs help, you can always apply for a proposal of a research on it. That I think I need to work to find out smoking causes cancer. So I need, and maybe nobody has ever worked on this problem until now, and you think that you're a pioneer in the whole idea, and then you can apply a proposal. You can do some literature research, go online, search PubMed, Medline, and you say, okay, nobody has seemed to work more on this problem. Then you can search more on the idea, make a proposal, draft it, and send it that I want to research on this. If it's a really problem solving proposal, lots of people will be willing to sponsor or willing to finance your project. Supervisors will be willing to pick up your project because they want their name associated with the big realistic problems. So it totally depends on you isolating a good real problem and work on it. You can do it. Pharmacists can do it, physicians can do it, nurses, anybody can do it. Then some people are specialized only for researchers. They only do some kind of basic biomedical sciences degrees. A biochemist might be involved, a molecular biologist might be involved, a geneticist might be involved, depending on whatever your background is and whatever type of research you want to do. For example, now Sam, she went to pharmacy. She's a pharmacist. She went to University of Calgary now, and she's working on pharmacoepidemiology in one of the projects, where they're talking about impact of marijuana and whatever the opioid addiction issues they had in the past. Because our work experience have been in the opioid addiction pharmacy, so it links her work with whatever proposal. She creates a proposal. She created a proposal, gave to her supervisor, random supervisor. One supervisor picked it up. They're financing her project. Now she's doing some masters, which will become a PhD from 2019 onwards. So the point is, it's automatically becoming a research, even though she's a pharmacist, but she's doing a pharmacoepidemiology kind of a study. So it totally depends on whatever background you're coming from and whatever interests your profession. If I was a geneticist, maybe I will try to look for some problem related to genetics, and that's how I can become a researcher. But then you cannot be an all-rounder. You will know only the clinical aspect of things. How do you know how to run a trial? How do you know how to do all that? That's where you get people which are called the they have names, trial CRA, clinical research assistants. And clinical researchers is the researcher is usually an epitome of it, which takes 30, 40 years to become a clinical researcher. But you have CRAs. A lot of people are clinical research assistant. And what do? They get some 1 to 2 years of degrees and diplomas to know how the whole process works. And then they become assistants. They help all the research go forward. And that's how you become in touch with different team. So the supervisor of a project will create a team: clinical research assistant, a pharmacist with clinical information, another person who will do documentation for us, someone who's an expert in journals, who knows how to write the format of a journal, someone who is a statistician join the team, who can do the stats numbers for us. So it's a whole team creation. Then the trials go forward. Okay.
And even the drug delivery, like drug
Drug making,
process,
yes. Drug,
same.
drug, yes. Same idea. Teams get created of different professions. They get together. Now, what is a cross-sectional study? It is also called as survey. Survey means I'm trying to survey out in the market what exists today as of current date, versus what existed 10 years ago or what is going to happen 10 years after. That is not my problem in survey. Survey is cross-section of today. What exists in the market today? The prevalence of a problem, not the incidence. Incidence means something which is new happening. What is the incidence of a new disease today? I can't say, looking at today's picture only. So when government wants to know how many males and females are in Calgary today, they're doing census, right? They go door to door or they do something which is called a survey. That is cross-sectional study. All they will find out is today's population in Calgary, I've got 500 people who have colon cancer. They cannot say what caused it. Was it there 10 years ago or not? They cannot say, will it be there after 10 years? They cannot say who was causing it, who was associated with it, nothing. So survey has limitations. These are the side effects. These are not side effects. These are the down aspects of the survey, cross-sectional study. But the good thing is, if you want today's data, you can get a good snapshot of today only. Okay. So what is cross-sectional study giving you? Prevalence. That what prevails in the market today, what is existing already. Does that make sense? Let's go forward and say what happens in a case control study. You have cases, you have controls. So cases are my people who have the disease. Controls are my people who do not have the disease. So I'm comparing two groups together. Similarly, I can have another population where I have cases where I am giving them the medication versus controls where I'm giving them a placebo, or just a sugar pill.
As it said, is there is a healthy people and the diseased people, or both of them is disease, or what?
Totally varies depending on what my goal of the study is. If I'm trying to identify effect of a drug on a disease, I might have both of them diseased. One gets the drug, other doesn't. If I'm trying to find out what causes the disease, diagnostic kind of study, One might have the disease, other may not have the disease. Then I'm going to try over time and see exposure rate. Let's take both examples and let me explain. This is the disease example, for example, and this is the drug example, for example. In the disease example, someone has lung cancer, someone doesn't have lung cancer. Then I expose them, some people to smoke, some people not to smoke, as in like out of my lung cancer people, some people exposure to, no, one sec, it can be in two ways, retrospective or prospective. If I'm saying in the past, lung cancer guys, those who were exposed to smoke, those who were not exposed to smoke. Similarly, those who do not have lung cancer, who smoked, who did not smoke.
Mhm.
Similarly, those who have lung cancer, who smoked, who did not smoke.
Okay.
So that's disease-based idea
Mhm.
versus drug-based idea. Some people have diabetes, some people do not have diabetes. I want to see if metformin is good for them or not. So I give them metformin. Some people I did not give metformin. Versus if I was to give them non-disease patients, I will not give them metformin, but just an idea, maybe placebo. Versus those guys who did not get nothing. Oh, sorry, this went bad. Let's start from the beginning. Diabetes guys who have this thing versus those guys who do not have the disease.
No, do you
Oh,
mean
those who did not get the drug. Sorry, my
yeah,
bad.
the
Is
distribution
disease situation is different. This is the drug situation, right? So let's from the beginning. So those guys who got the medication of metformin, those who did not get metformin,
Okay.
and all of them will have diabetes. This is the drug-based situation. So usually you'll have one of the two as a commonality. For example, drug or no drug, disease or no disease. So in a study, in order to compare, either I will have guys, both of the group having the drug with disease, or I will have a commonality of both of them having the disease or not with a drug. So it's like either drug will be common between the two groups or the disease will be common between the two groups at any given time. For example, if I'm trying to know the drug, both of the groups will have disease to test the drug on. If I'm trying to know the disease, then one of them have the disease, one of them will not. Maybe both of them might have the drug. So one commonality has to be there in order to cross references against the other one. But anyways, once we do more practice, you will get to figure out more and more. But the idea back again, what is case control? You have cases versus controls. Controls could be either without a disease, given a placebo, or they could just be anything, which is not the main case. Now, the cases are called the experimental group versus the control group. Experimental group means you gave them a medication, a drug, or you can say they were the ones whom you're targeting, your target population. So that's where any point in time, the experimental group, the case group, any event takes place. For example, an outcome happens. We call it the experimental event versus control group having an event. It's called control group event. And we'll go forward and explain more what exactly do I mean by event rate. Let's stick to an example. It will be able to help you understand the whole idea. Let's create an example, not eraser. Okay. Let's take smoke and the lung cancer as the example. Does that work? Does that work? Does that work? Yes, it works. Will be good. Let's do this. So I have a group of people who have lung cancer. A group of people who both the group of people have lung cancer. I have a patient population who have lung cancer, divided them into two groups. Some of them in the past who were exposed to smoke, some of them who were in the past not exposed to smoke. Now I'm trying to compare the two groups. What is my experimental group? The one who were exposed, where the exposure was positive. They are what? My experimental group. Right? Versus those who were not exposed to the smoke, They're what? They're my control group, right? They're the controls trying to balance out. Why did you control to think of it? That means if I have 5 people who have cancer and 5 people who have cancer, I'm just trying to see that could smoking be the reason causing this? Or is it just by chance, by luck, by coincidence, or by natural turn of events that they got cancer without blaming the cigarette or smoking? That's why you're having a control. If you have 10 people, and if you just say, did you smoke? 4 people said we smoked, 6 people said we didn't. That's not enough for me to say that smoking caused cancer. Versus if I have 5 people who smoked, sorry, 5 people of cancer, out of which 4 people smoked, whereas 5 people of cancer, 0 people smoked, then I have a reason to say, okay, only smoking is common with lung cancer in here, whereas smoking is not common with those who do not have lung cancer, for example. Right? It's just the idea of control is usually to just give the balancing idea. Here, both of the group people are lung cancer people. That example which I gave of non-lung cancer was just to explain the control idea. In this case, we have both guys having lung cancer. We divided them into 2 groups. One of them we said were exposed to smoke. One of them we were not exposed to smoke. That is one way of doing it. Alternatively, if I take 2 groups of people, smokers versus non-smokers, and then I say, okay, over a period of time, and now this is cohort I'm talking about, not case control. Over a period of time, I observed the smokers 10 years, 20 years, and similar demographic people, same age criteria, same other issues in life, everything. I found non-smokers, and 10 years, 20 years onwards, I followed them over a long period of time. This is a cohort study. So after some time, I found out after 20 years that 3 out of 10 people developed cancer. For example, here maybe I got 1 out of 10% developed cancer. The idea is, if I was to just look at this part, and for example, I did not have my control group, for example, if I did not have my control group, how will I prove to someone that over a period of time, 3 out of 10 developed cancer because of cigarette? What if they will say, okay, well, anybody else could have gotten it too? But now I have proof to say, no, not everybody. Only 1% coincidentally. So maybe 1% was by chance. But what about patient number 2? What about patient number 3? 2 more patients got cancer here. So there's a reason to believe smoking might have been an association with cancer. That's why you need a control group. Does that make sense?
Yeah.
Just to give you a comparison, just to give you a rationale to prove or eliminate the chance. Because 1% happened by chance, for example, or because of other factors which might happen in life. Maybe pollution on the street, maybe genetic factors on the person, etc. So if you say 3 minus 1, I got 2. So I can say, okay, by some kind of 20-year study, I can say some people got cancer maybe because of smoking, right? That's what the idea is of the control. So case control study, you're doing it over a period of time to compare, to show that these causations or associations can be made some kind of relevance. Okay. We're going to study about these 3 in the text as well in the coming slide. RCT, on the other hand, is randomization. You pick a population, you randomly choose them from different groups. Who goes here, you don't decide. Only your software or there are different methods to randomize them. That's how you find it. They're assigned to different groups. Why are you doing randomization? Let's talk about that first, then we'll talk about levels of blindness. So, and then we're going to talk about the word bias. So randomization means randomly selected for different groups, right? And you're doing this in order to eliminate a possibility of error in the results. That means if I was to say that I will choose my own population for group A versus group B, if I'm the study researcher, I might have some favoritism towards my research. In order to get a good result, I might do that, which we do, which we still do, right? That whenever you present a research study, you cut the corners, say, if I present stats this way, it will look really good. For example, when I was doing my master's thesis, I had double thesis in my master's. One was about interventions in clinical pharmacy. So what we did, after trying to find error rates, medication errors of pharmacovigilance, error rates in ICU units for parenteral drugs in the university I was working at, after almost 600 to 700 forms filled in, self-reporting forms, the error rate was reported to be 0. Not even 1, 0. I looked at it in all angles how I can present it. I can't present it. If it was 1, I can do something. It's 0. Then I just presented demographic data of my patient population, somehow went through it and did double thesis on it so that I can have alternate ways to look at it. Point being, you cut the corners. You do whatever you can. I took specifically 1 week to figure out cheating ways, how I can make my thesis of my degree. Then I spoke to my professors. I don't know. I can't do anything. He said, no, you have enough data. Just present it. So I just did it. So the thing is, randomization is to take that favoritism out of the picture, that human nature out of the picture, the researcher's bias. Then there might be unintentional bias as well. You don't want to do bias, but naturally it happened. That, okay, I went to Calgary and I said, okay, Calgary group in A. I went to Edmonton and said Edmonton group in B. But later I found out, okay, there were weather differences in the two places. There were genetic differences in the two places. Then the results got error. They're not correct results of the study because both the groups officially were different. So that's where you take a main group and randomly assign them in different groups. The bigger the population size, the more reliable your results. Because if you take 2 patients, one in this group, one in that group, and work on them, that chance possibility might kick in. One of them might have cancer. He might die in a car accident. Your study is done. So you need bigger population to get better results. So randomization is to eliminate the bias. What is bias? Error. There are different types of biases. One is, of course, selection bias. That I want to select this guy into this group. So you eliminate selection bias by randomization. Easy. Another type of bias would be recall bias. For example, if you're doing a retrospective study, let me define the terms. Going backwards in history is retrospective study. Going forward is prospective study. Retrospective means I'm sitting today and digging down into old data. I went to university research and found the last 10 years data of people with colorectal cancer. I found something this, or maybe last 20 years. This is digging down past data, retrospective. Versus prospective would be a cohort kind of study where I'm sitting and saying, okay, I'm going to observe this group and I'm going to see how it happens in the coming 5 years on these patient population, how the effect of this medication is going to be on the coming time. So retro means looking back, prospective means looking forward. Okay, so recall bias could be a problem with retrospective study. For example, I go and talk to a patient. Hey, 10 years ago, you were having a problem of insomnia, and my report from the hospital says that you came and reported this problem and you got medication treatment. The guy's like, yeah. How many times did you sleep? I think it was 5 hours, maybe 4. This is what he's recalling from his memory. And that is going to not bring you the real data. Because when you made the data 10 years ago, a patient came to you, what is the problem? I have insomnia. You just wrote insomnia. You didn't say he slept 4 hours a day, or he took this medication, he tried antihistamine for 3 days, didn't work, he didn't try sleep hygiene. No data is there. It only says he had insomnia and you recommended zopiclone and he went home. He never came back. So next, you don't have any more data. So now when you talk to the patient, you get recall bias. For a smoker, you will say, for 3 years ago, you were smoking. How many cigarettes do you used to smoke? Maybe 2, maybe 6. Sometimes this, so he's not sure. No official real record is recalling. That can bring wrong results because it's not real data. So that is recall bias, right? There could be such types, many such types of biases involved in the study. These are some important ones which we've spoken about. Okay. In order to eliminate that, you need authentic data. Then let's talk about blindness. You blind, that doesn't mean you pop the eyes out of the person. Blindness means the person doesn't know what's happening in the study. Okay. So there are different levels. One, your patient population don't know which group they're assigned to. Are you getting the medication? Are you getting a placebo? They don't know. You okay?
I'm just bothering me with the sun in my eyes. Can I do it?
Yeah, sure. Is sun leaking through this somehow?
Yes.
Nice. Water and light, they find their way.
So can we say the cross sectional study is a retrospective study because we are
No, it is current
the
of today.
cross-sectional.
Cross-sectional is today. It's a good question because cross-section is right in the middle, survey. Retro is backward, prospective is future, current survey is today. Whatever exists in the world today, right now, at this moment, is what you're finding out. Retrospective means what happened 10 years ago, what happened 5 years ago, 15 years ago, that's past. Does that make sense?
Yeah.
And sorry, go ahead.
So the cohort study is the future study, which is prospective study.
Well, to be honest with you, cohort, I gave an example of cohort itself has two types: retrospective cohort, prospective cohort. But you don't have to worry about it. All you need to know is case control usually is either retrospective, usually retrospective case control, whereas cohort usually is future. Usually
What? Case control, what you said?
back, retrospective,
Or today?
past,
Case control.
case control, usually past. But it can be future too. Can be future too. I'll explain all of them, don't worry. Cohort usually is supposed to be a group of people you follow up over a long period of time, usually prospective, but sometimes only retrospective. And that's where the confusion is between cohort retrospective and case control, because you're looking back in both of them. But you don't have to worry about it. For exam point of view, at this point in time, and as we build the concepts forward, I promise by the some point in December, you will know all of them, which direction they go in, and you will know exceptional cases also. But right now, I will skip the exceptions. We'll talk about the general rule and then build the concept, and exceptions we'll build on it later when we do MCQs practice and all. So general rule, case control, retrospective, cohort, prospective. Okay. Survey today, current. No retrospective, no prospective. Prevalence of today. That is what? Survey, cross-sectional. Okay. So we're talking about blindness. There are different levels of blindness. Single blindedness means patients don't know which group they were assigned to, whether they're getting the medication or the placebo. This is the basic level of blindness. So that if you tell a patient you're taking placebo, he'll start skipping it. So there's a placebo effect as well. When you take a medication, the patient thinks, oh, I'm going to get healthy. I got the medication, right? There are times you just give them sugar pill and they say, doctor, I feel so good after the medication. That was a magic pill, but it was just sugar pill. I don't know. It is what? Psychological effect, placebo effect as we call it. So patient is not knowing. Second level of blindness would be that the administrator, the guys who are giving pills, pharmacists, doctors, we don't know which pills they're taking. Are they getting assigned to the placebo group or the medication group, so that we don't have any bias involved? Because if we know which group they're in, we may have different kind of medication therapy for them. For example, a guy with diabetes giving metformin, the other guy with diabetes getting a placebo. And me as a healthcare provider, I'll be like, the guy who's getting metformin, okay, take it with this and take with food and whatever not. The guy who's the other one will be like, take this pill whenever you want, but make sure you take no sugar diet, okay? Make sure your exercises are high, okay? What are you doing? you're biased right now because you're trying to help the guy out. You know he's getting a sugar pill. He's just getting a placebo pill. So there might be difference in your therapy, in your care. That's why you're blinding yourself here as a healthcare provider, blindness, so that you don't have any bias involved. You treat both the groups same. Third level of bias is even the researcher doesn't know. Even the researcher doesn't know. Triple blindness. That means patient, healthcare professional, and the researcher, they randomly assigned, and only it's hidden in certain ways. Usually, it's the owner of the research only who will know. He'll be provided randomization details, and at the end of the whole study is done, then they tally the two and say, okay, these patients who died were the ones in placebo group. These who survived and doing amazing life are the ones who got metformin. Okay, so that's the difference you got from the end of the study when you tally and open the envelope and say, oh, that's what happened, right? That's when you tally. That is what when the researchers are blinded as well. Okay, the highest level of evidence here would be a randomized control trial with triple blindness. Very high level of evidence. That means reliable study, no bias, no recall bias, no selection bias, no healthcare professional bias, no researcher bias, etc. Right? Are we clear on the idea? Good. Let's read a little bit. Cross-sectional. The survey. Collection of data after disease and exposure is already done. So you're just doing what? Collection of data as of today. What is the advantage? It's fast and provides prevalence. We discussed that already. Disadvantage, based mainly on self-reporting, that you go and ask them, do you smoke? Yes, I do. There you go. Bye. Then you go to the next person. Diagnostic information usually inaccurate, not reliable info, does not establish causal sequence. You're trying to create some kind of causation, but this guy doesn't give you causation. It's just a snapshot. Case control studies population with pre-existing disease and one group without disease, for example, the control group, right? To determine what? The potential cause. That's now the advantage of case control. It can help you find some kind of causation. Advantage, sometimes you can catch a rare disease in this. It is fast, relatively inexpensive, and don't worry about odds ratio right now. We'll talk about it in the coming slides. You calculate odds ratio in this one, whereas you calculate relative risk in the cohort. And as I said, don't worry about these two right now. We'll talk about in the coming slides. Now, disadvantage of case control is difficult to select the control. Bias from the patient recall. Exposure status relies on records and recall. So there's a lot of bias because it is based on case control. We said is mostly retrospective, back. So you're asking them, did you smoke? Did you sleep or not? All that. Recall bias. Relying on the records is bringing bias, etc. So these are some disadvantages of case control study. Now let's talk about cohort. Studies a group of individuals over a long period of time to establish the time sequence in which things occurred. For example, in 1987, let's say 30 years ago approximately, new graduates of a huge nursing university, they recruited nurses, the new nurse graduates, as their participants of the study. It's a huge nurse study, and now there were more than, I don't know, let's say 3,000 nurses involved. Until today, we're doing research on them. They're still participant of the study. Since they're healthcare professionals, they're able to comply a little more than general population. So still there are approximately 2000 or 1800 nurses still in the study enrolled. So since almost their youth all the way to their mid-age, beyond mid-age, we're testing on them, not testing on them, we're following up on them. So many kinds of things that if this happened, then what? They were having some kind of pregnancy issues. They might have breast cancer development, etc. So lots of endpoints were calculated there. So it's still going on. The large nurse study. Okay. So that's what cohort. You picked up a group. Cohort generally means group. Sometimes they will ask you, which cohort do you belong to? Oh, I belong to that class of 1997, for example. Cohort. Cohort means group. So group of people over a period of time, you're calculating things on them, doing testing on them. So studies a group of individuals over time to establish the time sequence in which things occurred. That some of the ladies died in 2002 because of this. When they were exposed to some kind of smoke, this happened. Or some of the ladies in Singapore, they were exposed to asbestos, they got some kind of cancer development after 15 years. Long story short, long duration. Advantage, it can establish causal sequence that only those who started smoking started developing cancer. It can give you the incidence of the new disease. Incidence means what new incident happened. Incident means an happening, an event taking place. That an incident happened on the Northeast today, for example, that means a new event happened there. So over a period of time, we can say a new disease started existing, a new happening happened, versus survey or cross section just gave you prevalence, what exists already. Here, you don't care about what exists already. You want to know what a simple problem is going to cause a simple disease. Causation, incidence rate. So that's what this guy is giving you. Whereas case control doesn't give you incidence, doesn't give you prevalence. Neither of the two. Cross section gave you what? Prevalence. Cohort gave you what? Incidence. Case control gives you none of the two. Okay. Disadvantage of cohort is they're long and costly. They're still spending 30 years on the same group. Participants may withdraw. It's not easy for them to stay in your study. I don't recall what I was thinking 10 days ago, let alone 10 years ago, right? Like 10 days ago, I was saying, I want to eat this. Today, I feel like not at all. I hate that food. I want to eat something different. Natural. Your change as person change, right? You grow older, you become wiser, a lot of things change. But not easy to be part of cohort study over a long period of time. That's the whole bottom line. Okay. Want to take a break? We'll take a break and then we'll talk about. It's a short break, not the lunch break, just a coffee tea break. Let's say it's 11:17. We'll come back at 11:30, and then we'll start again from where we left. Okay, so these are the types of studies you need to know. Some of them are already mentioned. Case reports, we already said what case reports are. Individual reports. Cross-sectional is a survey. We already said that. Case control, case versus controls. Cohort is a group followed up over a long period of time. Now, this is a new term called quasi or quasi experiment. To be honest, you don't need to know this much, but just the name quasi, whenever you see, you should think of non-randomized RCT. Because RCT is supposed to be randomized control trial. A non-randomized trial is quasi. Okay, that's the only thing you need to keep. Not randomly assigns groups, and you don't need to know more details. It's not in your scope of study, but it's just that if you ever come across quasi, you should know it's non-randomized. RCT, we already know. We said it's the good level of randomized control trial. You do random assigning, blindness, whatever. Then comes two important things at the top of the whole mountain. One is meta-analysis. We already discussed a little bit about it. The second thing is called as systematic review. So they're two different things. And to some extent, some overlaps too. And they are the best type of evidence you can make. Most reliable literature I would like to search. Number one. Different RCTs exist out there. For example, they have done some kind of good studies, or maybe there could be one case control study, some cohort study, etc. So I found some really good studies, and I said there are two researchers I have, for example, and I gave this set to one researcher and this set to the second researcher, and I told person A that, you know what, since you're a biostatistician, I just want you to pick up only the statistical information from these studies and present the statistical part of it. You're not a clinician, you're not a pharmacist or a doctor, you don't know the clinic aspect. Just give me the statistical analysis. He did that. That is meta-analysis. He just took the statistical part, right? Without interpretation, without analyzing it, without giving his input. It's not needed from him. Whereas another guy who's a clinician, a doctor, prescriber, or medical or pharmacist, we told him, okay, you know what? You take these studies and give me a review of it together. That means I want you to give your interpretations on the 4, 5, 6 studies together and give the compilation of a result for me. And I want you to say that, describe that if these 5 studies used metformin, I want you to give me a conclusion of sorts. That is one way of making a review. Did you get the difference? This is systematic review. Systematic review. Because you took several studies and combined them together. Several good quality studies, and then you combined them. Each systematic review will have its own criteria, that no study should be below this level, or all of them should be RCTs, all of them should be randomized, I need double blinding, or all of them should be triple blinding, etc. Criteria could be lenient or strict, varies. A company named as Cochrane, it's an institute. They are known to do very strict criteria of very high quality randomized trials. They don't do trials, they take information of trials, very high level of blindness, high level of randomization, good study designs. They make both meta-analysis as well as they do reviews on them. They have teams. One exception though, sometimes a systematic review guy, the clinician, might come and say, yeah, you know what, I'm still going to review the articles for you, but can I get some statistical information as well? We'll say, yeah, sure, my another colleague meta-analysis is there for the same studies, you can use that as well. He can use some statistical information compilation from the other colleague, and he can take the real 4 or 5 studies as well together. He can give a good review. So sometimes systematic reviews may have a meta-analysis within them, right? That's what we say, systematic analysis, systematic review based on meta-analysis, okay? So these are basic differences between two. When it comes to the level of evidence, which one is more reliable, a general meta-analysis or systematic review? To be honest, meta-analysis is more superior than systematic review.
This one is just observation, just
This is just
collecting the
statistics.
information. Not
That's
really.
right. Because one of the advantages is if I give a meta-analysis, some study to you and some study to another person, you both might be able to give me different interpretations, right? Two different interpretations, reviews happen. Now, when I'm sitting in the pharmacy and I'm trying to apply some knowledge to a patient, I look at two different reviews. Then I'll be like, I don't know. I might as well go to the real study to find real information, raw information, and interpret myself. Because right now the reviews are different, interpretations are different. Maybe I don't agree with your interpretation.
Yeah.
Maybe I have different opinion about. So that's where a meta-analysis is raw statistical information. Interpret the way you want to. That gives you a reliable information, eliminating the bias of a human person from the middle. That's where meta-analysis is above systematic reviews number 2. These are the top 2 in the whole pyramid, as we say. Do we have pyramid coming soon? This is the pyramid. Meta-analysis is top of systematic review. This is the level of highest to lowest. In your book, I have written meta-analysis is more than systematic review, is more than randomized controlled, like that. It's the same idea. Meta-analysis, number 1. Systematic review, number 2. RCT, number 3. Now, another thing is you can call these ones as non-randomized as well. Because randomized control trial, non-randomized control trials. Actually, these two are with the control. Sorry, because case control cohort has a control as well. Do you get the idea of the pyramid I'm talking about? Reliability on the strength of evidence. So let's go back. So I hope all the ideas are clear on the different types of studies. Whenever you see a study, I hope you'll be able to recognize it. We have some examples as well if you want to practice at your free time. I have 6-8 different case studies written. You can see and say, okay, which study design is this? If you're able to pick up a study design, I have answers at the back, so you can figure that out. It'll be good practice to know. Cross-sectional gives you what? Prevalence or incidence? Prevalence. Case control gives you what?
No,
None of the two.
No prevalence, no.
And cohort?
Incidence.
Good. Then, when we do calculations, we need to find out which one would be falling which. So at this point, just for memory, odds ratio is the calculation we will use for case control, guys, whereas relative risk is what we'll find for the cohort, guys. Probably your book might have risk ratio as the word there. Is that right? That is just an abbreviation of RR to make it easier with odds ratio. But the real RR means risk ratio, not relative risk. Sorry, it means relative risk, not risk
It's
ratio.
just a relative risk.
Maybe that's the corrected version. But some of you might have risk ratio. You have risk ratio? So correct it to relative risk, please. You were looking for something there? You got it?
No, I'm good.
Okay. So randomized control trial and meta-analysis, we've already defined them. Okay, now there are few. Actually, you know what? I'll give you more printouts of all these too, because there are several things coming which are new than your book, right? I'll give you all printout as well.
Thank you.
So 3 definitions: epidemic, endemic, pandemic. This is also new. I added this 2 days ago. Endemic means a disease which exists in a place. For example, malaria is a constant worry in parts of Africa. So what is that? That is an endemic. Within that place, it stays there for a long period of time. Versus, what is epidemic? An outbreak of a disease. Influenza outbreak is epidemic nowadays. It's epidemic, very normal term. An outbreak of disease that attacks many people at about the same time and may spread through one or several communities. Like Northeast had having influenza outbreaks and it became to Southeast and downtown, etc. Versus a pandemic is a way bigger version that a disease took over entire Calgary or went all over Alberta. Pandemic, it's increasing. Ebola virus, for example, it's a pandemic then. HIV is a pandemic, right?
Endemic, what's the difference? Endemic, it means for a long time.
Endemic means within a small area it stays there and it's not spreading out to more places much. It's just confined in that particular place and stays there for a period of time. That is endemic. End means inside. Epidemic means contagious, spreading out. Epidemic means small areas spreading out, whereas pandemic means bigger areas spreading out. So epidemic, pandemic.
Okay, what is the difference between endemic and epidemic?
Okay, epidemic, as I said, is spreading out more. Endemic is staying within confined area.
Okay, but same diseases doesn't matter. Like for example, flu. Like if it's only in this room, so this is endemic. But if the same flu go out to southeast, now it's epidemic.
No, this is a good question, but here's how you want to define. It depends on the nature of the disease. Malaria is confined to those areas where there are some mosquitoes, there are some places where that is more possible. That's why malaria is a constant worry in some parts of Africa and it stays there. It may come from there and person might bring it here too, agreed. But there are other ways where malaria will not sustain here. Probably that cycle between hepatozoites, hypnozoites, as well as in the life cycle of mosquito, that life cycle will not be maintained. It will stay for 1%, maybe few days, few weeks, and stay and die it out. But then malaria wouldn't spread to others much. This can be very highly likely possibility. Whereas if someone is in Africa, it stays there in that area. Whenever I will go without having malaria, I go and will catch malaria, right? Because that area itself has endemic. It is within that area, confined. Versus influenza, if I catch it from there, if I come here, I can give it to you, to somebody else will give it to their family members and others. It is spreading. This is epidemic. Does that make sense?
Yeah.
It depends on the nature of the disease. Malaria is not spreadable much. It stays in that area much because of the factors which define this disease spread. For example, some of the diseases which are more hygiene related, some of them which might be like, for example, typhoid. Typhoid stays in a place. It won't be epidemic. It will be an endemic because we can say
The environment.
one environment, one city in let's say in Pakistan got uh, typhoid fever. A lot of people are having typhoid fever there because of the spread of the bacteria, because they were drinking maybe similar water resource, maybe they were probably having similar food resource, etc., right? So it's confined in that area. If I go there, I might catch it, but those people are staying there, they will have it. If they come here, they may become healthy, and then after that they may not have the further recurrence or further infection. Someone who stays there keep on getting that infection again and again. Why? Because it's confined to that area. Endemic. Agreed. Influenza, on the other hand, is epidemic. We get it, but after a few weeks, few days, it goes out, it fades out because it's epidemic, starts spread a little bit, then stops because people get recovery, they get flu shot, they get medications, etc. Recovered. Pandemic, on the other hand, is a bigger
Sir,
version.
what? Sir, what about pneumonia?
Pneumonia.
Pneumonia,
Yeah, pneumonia. Is it pneumonia? Is it epidemic or the
good.
uh,
It's considered epidemic, to be honest. But you know what?
epidemic?
Contagiousness
Yes.
of pneumonia is negligible. Not negligible is a bad word. Is way low. It's very
Okay.
less. Pneumonia contagiousness is very low. The reason for pneumonia is it has to be more host specific factors. Host or the person who getting infected should be very receptive, or should be very, environment should be very conducive for the bacteria to grow.
Okay.
Immunity levels are low, other factors are there, integrity of the lower respiratory tract is very damaged, etc.
The last question I want to
Thank you.
answer. We have in Egypt the disease for hepatitis viral C, and this one is epidemic or endemic now. This is
Hepatitis C would be considered as pandemic. Hep C can go all across the world. Lots of Hep C people are here as well. Again, it's
But
specific idea.
sorry,
Yes,
it's depend also on the gene of the virus, because it's for example in Canada it's more genotype 1, and in Middle East, for example, genotype 4. So this is could be depending on the virus protein
that
also. So
agreed. That is very good info. That's true, Ibrahim. But when it comes to endemic and pandemic, that's irrelevant. The idea is whether the hosts are receptive to the virus or not. Different story. But if a person who have Hep C in Africa, or if have Hep C in any Southeast Asia, and it comes here, he can spread hepatitis C for other people as well. Reason?
can do it.
Because it's a multicultural country. The whole world has multicultural environments now. That doesn't mean the Caucasians are the ones who are going to get infected too. Maybe they may not. They have different genotype. Agreed. But a lot of people like you and I are coming from different world countries. Our genotype is that. So we still can bring Hep C to Canada and give it to each other rather than a Caucasian person or a Chinese person, for example. So the point is, it's irrelevant what the genotype of the background is. Pandemic, endemic can, yes, genotype can limit the pandemic's growth, but eventually Hep C is something which is highly virulent, spreads, contagious, and it can be big.
So you mean like the pandemic, like the HIV? It doesn't matter how many people they are get that virus in the other countries. Like for example, overseas, like for Middle East. it's not really that much spread.
Yeah.
But here is a lot. But it doesn't matter. Still pandemic.
Yes, it is pandemic because it's spread across the world. It's way bigger than an epidemic. Epidemic is limited to a small area. We already discussed the hierarchy on this thing, which we call as the pyramid, the evidence pyramid. Okay. Meta-analysis one case. Actually, if you want to add something at the bottom, it is called the expert opinion at the bottom. That means case report is just one report, but the idea is it's still a report. It's one report, but at least there is one person's case you're presenting. Expert opinion is just that I think this should be that way. It's just your opinion, buddy. Who cares? It's the least evidence. If I'm practicing in a pharmacy today, and if I was to give some medication to someone, I want some strong, reliable information to back that up. I can't just simply say because some expert thinks this medication is good for you, I'll give it to you. No. I want some reports. I want some trials. I want some evidence to prove it strongly. Rather than just because Mohsin thinks so it's a good drug. Who is he? Nobody cares. What matters is the evidence. So that's where the reliability of evidence has to be more. So expert opinion comes even below a case report. Okay.
Terrible.
So off label drugs, which column consider in this systemic or randomized control?
Off-label drugs has nothing to do with this idea. It's a very different concept you're talking about. The
Okay.
idea is any drug can be off-labelly used. Doesn't matter the trials they came in.
Mhm.
But he means this is the off-label drugs used in another case, like or to treat another thing, not in the pamphlets or the
Right. So that is not based on much. That's not based on any specific criteria which study it came from. It could have been a case control study, and you said, okay, if you were using metformin for eye infection, it helped. Okay, off-label use, maybe it can happen. But government says I don't want to prove this until I see really good studies. Until the approval has happened totally, physicians are still prescribing it because they think it helps their patients. Off-label use.
we can consider it in phase three.
Factory.
Phase three.
Phase three. Okay. Yes, that is where you can think of it.
Mhm.
Phase three. Any study which might be in phase three. No, 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 no. Oh yes,
Because
sorry.
this is
Yeah, you're right. My bad. If a drug is in phase three, and for any reason it showed some benefit, and usually off-label usages are accident.
Mhm.
Off-label usage drugs are accident. For example, there are people who have diabetes and you are using metformin on them. There's a trial going on of metformin, and suddenly you realize that all of those guys who are using metformin, they were having very good body weight, BMI, eventually, after using for a year or so. Good BMI. Then you started noticing, is there a link? Is there a possibility? Then you figured that out, and some doctors are like, you know what, we found that out in phase three of this study that this medication is doing this. When the drug comes to market, people are using it for diabetes, and some physicians will say, okay, you know what, it gave a history in the past, it has done some kind of help to weight loss. If you are really obese, use metformin. Maybe they might do that off label use, and they do it, by the way, metformin for off label use. So phase 3 and phase 4 both off label drugs can come from there. But when it comes to what type of study, Ibrahim, it could be not meta-analysis, it could be anything, any kind of study from which the data is being taken out to decide off-label usage. Any of the study. Okay. Now, when you make epidemiological studies, you need to do something called as 2 by 2 contingency table. This table is called as 2 by 2 table. That means you make a chart or table, A, B, C, D, 4 squares. Okay. Then on the north, on the top, you have to put the outcome. On the left side, on the west, either the exposure or the tests which you're doing. Outcomes of those, either the exposure or the test, will come here. So let's put in some examples one by one of exposure and a test, and then we'll make sense out of it. If we're talking about cigarette burning,
It's like petrol or something.
maybe they're making the road. Yeah. They finished the the parking lot since about a few weeks. I was saying, thank God they started some new project. So exposure, smoking and lung cancer. Let's take the same example. It makes it easier. Exposure is the idea. What is the exposure? Smoke. Smoke. If you're exposed to smoke or not exposed to smoke. So let's say if you were exposed to smoke, that means you're positive of cigarette smoke exposure. Negative, that means you were not exposed to cigarette smoking. Then outcome means lung cancer is the outcome. You have the disease and you don't have the disease. Okay, this is what you put. Now, out of those people who smoked and who actually got the disease are here. Out of those people who smoked and did not have the disease are here. Those who didn't smoke and still have the disease for whatever reasons, they're here. Those who didn't smoke and don't have the disease are here. And this is how is the exposure idea. Let's look at the tests. If you want to put some kind of a test, for example, urea breath test. You know what urea breath test is? You do that to check the peptic ulcer disease. Urea breath test is checked because urea is being produced by the bacteria called H. pylori, and you're going to work on it. So now you're saying that in order to catch the disease, I'm going to use a specific test. We call it a screening test. So UBT, urea breath test, screening test. Now we're saying those guys who tested positive, those guys who tested negative, or they have the disease PUD or not. That is the outcome. So if my people tested positive, those who has the disease will be written here. Total number of them, maybe 5, 10, 15, whatever number. Those who don't have the disease and they still tested positive wrongly, that is here. Those who has the disease and they tested negative wrongly are here. But then those who didn't have the disease and they tested negative are correctly here. Right? This is just an idea how to put tests versus the exposures in the 2 by 2 table. Now let's look into the next slide and see what are these. Let's look at the UBT idea again, urea breath test. Positive, negative. They have the disease and they don't have the disease. So those guys who have ulcer, and I want them to go and do the urea breath test. We did the test, it was positive. Makes sense, it's logical. They had the disease, it came positive. This is what true positives. They really have the disease and they tested positive, true positives. Now let's talk about those guys who didn't have the disease and they went to the lab, they did not get any result. No result. They didn't have the disease, it showed that they don't have the disease. Good, they're true negatives. True, because those who have, those who don't have, the results came the way they expected. But there will be possibility of having false results, right? So those who does not have the disease and they test positive, and then we give them the bad news, oh, you have the ulcer, and then they say, really? And then they start getting treatment, it turns out they never had the ulcer. False positives. Falsely we told them they're positive. Versus those who has the disease and they tested negative, and we said, you don't have the disease. They keep trying all therapies and say, nothing works because you have H. pylori. We're not treating that because urea breath test didn't catch you, right? So, ideally speaking, a good test will have good true positives and good true negatives, and a good test will have low false positives and low false negatives. Make sense?
Yeah.
We need the true numbers more and we want the false numbers less. That's where the idea of sensitivity and specificity of a test comes in the picture. We want to know whether a test is more sensitive or a test is more specific. So we'll do that in the coming slides. Let's finish the basic idea first. So are we good with the idea of where to put true positives and all those guys? Now, I want you guys to memorize the idea that outcome comes on the top, exposure or the test comes on the left. Okay, then you have to identify true positives, true negatives like this, and you have to identify all the positives are on the top so that you can easily, because usually what happens, people are able to put these easily, true positives, true negatives, then they flip the false positives, false negatives at times, which is going to flip all your results. Keep in mind, all the positives are on the top, TPFP, both of them are true positive, false positive, positives are on the top. That will be easy for you to put the fourth one here. Some of you in the book, if you have the previous version, you might have the flip model, you might have outcome here, you might have exposure there. Eliminate that idea. We're not going to go with the flip model anymore, because sometimes you will have issues. We're not going to go with that. We're going to stick to the idea of this one. This is your take home message. Over. Okay.
So can you explain true negative?
True negatives, that I don't have the disease. I had some GI symptoms, went to do the UBT. In the breath test, they said, negative, you don't have the disease. Okay. Truly negative, which actually was negative, and I found the true result. Make sense? Okay. You are making that one?
Yeah.
I will print this out for you at the end of the day.
Okay.
Plus, I will upload it on the website as well.
That's okay. I'm good.
Okay. So now we're putting all this in perspective. We already said that outcome is on the top, disease or no disease. Test score is here, positive or negative. We know where true positives, false negative, positives, false negatives, and true negatives are placed. Now you know how to make 2x2 table. Let's move forward. I'm going to define few terms. You don't have to memorize them yet. At the end of the thing, I'm going to tell you what you need to take home. And this is just concept building right now. And don't get overwhelmed with a lot of information here. First things first, there's a thing called positive predictive value. That is this square and this square. TP is A divided by A plus B. TPFP. So A plus B. This is just a positive predictive value. Even though a similar formula will be overlapped with another thing called as experimental event rate, don't worry yet. We'll come to that later. This is just a thing called positive predictive value. And again, we're just building concept to give you an idea. Second thing, there's a thing called negative predictive value, square C and square D. Tn is D divided by Fntn, which is
D plus
C
C.
plus D. There's a thing which we discussed a little earlier called sensitivity of a test. Sensitivity means A divided by, so we're talking about true positives divided by a total of all the guys who have the disease. Specificity is false positives, true negatives divided by all those who don't have the disease. This divided by this. And these two squares, A and D together, give you accuracy. A plus D divided by the total, grand total of 4 squares. There are so many terms. We spoke about 5 terms. Positive predictive value, negative predictive value, sensitivity, specificity, and accuracy. Now, what you need to know is at least about sensitivity and specificity. The other terms, not so much at this point. So let's talk about sensitivity first. It's a big concept, and I'm going to put it in some examples, try to explain. And if you're able to take it home today, I'll be very happy. What example should I use? Door. Okay. So, for example, in my house on my door, I have an alarm. For example, there's an alarm on my door. In the night, I turn the alarm on when I go to bed. If a thief tries to come, the alarm rings, right? And when there is no thief, alarm shouldn't ring. This is the way, the ideal way it should be.
Work.
Hmm?
Work.
That's right. But if I make the meter of the thing too sensitive, then a thief is not there, but even if the wind blows, a cat crosses, it starts ringing. That means too sensitive, catching more than it should, right? So if it is too sensitive, then I have to get up every few minutes and see the door. It is wastage of energy, wastage of time, money, effort, so many wasted. Put it in the medical perspective. If a test is too sensitive, let's say your breath test, if it is too sensitive, it's not only catching those guys who have the disease, but it's also catching those who don't have the disease,
How
right?
it
The wrong ones.
how it will catch not have the disease instead of there is no bacteria or something like that.
No, the idea is it is not sensitive test. It can give you false results because of any other factor. A cat, a wind. These are examples in my case. Now, in the medical context, you might have high level of uric acid in your body, for example, or you might be a smoker, for example. You might be on keto diet, for example. All this can bring urea levels up. Maybe breathing will give you urea test positive. Who knows? Some false positives. So a test is too sensitive, it over catches more than it should. That gives us what? Loss of money, loss of medical resources. Does that make sense so far? Now let's look at the flip situation. If I make the test or my alarm, let's talk about the door, too less sensitive, that means too specific on the opposite side, then it, so that the wind blow catch or the cat crossing doesn't ring the alarm. But if the thief comes also, still the alarm will not ring. The opposite situation. So I'm sleeping and the thief comes, steal everything and go, and the alarm didn't go. Right? This is what? Very less sensitive test. More specific. Sensitivity goes low, specificity goes up. So too specific test. That means it will not catch even the disease. It will catch very less people. It will eliminate most people, but it might still miss out on the real disease people as well. Right? Those people who have the disease, they might go home and say, hey, I don't have that ulcer. Right? That is what? Too specific. Did you get the basic idea? Now, let's put it on a little bit of a bell graph. The second one on the right side, this one I just made, is let's say too specific side of my switch. If I make it on the left side, it becomes too sensitive side of my switch. This is my meter changing. If I want to change it to more sensitive or more specific. So this is sensitivity and this is specificity side. If it's on the more maximum right hand side, that means it is too specific. This line which I created on the right, this is the distribution of patients, for example. On the right hand side are all those people who really have the disease, and we catch them, the true negatives. That if my test is too specific, that is on the right hand side, it is going to eliminate those people who really don't have the disease, but it will eliminate even those who have the disease. Remember we just said that even if you have UBT test negative, you might still have the ulcer, and you went home and said I don't have ulcer. That means you're catching only those who really have the disease and eliminating them, but you're eliminating sometimes those as well who have the disease. You're undercatching than you should. In sensitivity, you're overcatching than you should. Remember, you caught more people than you should. In specificity, you're undercatching than you should. Now let's look at the flip situation. If I create a line here, I am eliminating those guys totally who are true positives. That means sensitive test, too sensitive. I am even catching my thieves. At the same time, wind blows, my alarm rings. Too sensitive? In medicine, my urea breath test catches people with ulcer. It also catches those who don't have ulcer, right? Overcatching. So the problem is, on the left hand side of this graph, all these guys are actually having the disease. We caught them the true positives, versus there are guys who are lying in the gray area. That means my false population of false positives and false negatives lie here. False positives and false negatives. Now the idea is that gray area, how can you identify that? That is the problem. True negatives are easy. You don't have the disease, test came negative, go. You have the disease and we found a test positive, come and take the treatment. These are easy groups. But what about false positive, false negative population? How do you decipher? How do you differentiate it? It's not easy. It's not easy. So there is usually a thing called trade-off. Trade-off means, okay, in order to have a test more sensitive, I might lose on some specificity. In order for a test to be more specific, I might lose some sensitivity. So it's a give and take. You cannot have the best of both the worlds. What would be the best? If you have a test right in the middle. That means on the right side of it is total specificity, on the left side of this is total sensitivity, which is hard to achieve in any tests. It's very hard to achieve the balance.
It means no gray area.
Yes, it's almost impossible to achieve that through a good test. So what do? You say, you know what, since this is a problem, let's have two levels of testing, a screening test and a diagnostic test. So we will set their sensitivities and specificities in a way where we save the cost and at the same time we catch more people when they deserve to be caught for the disease. Getting the idea where I'm coming from? You don't have to memorize all this. This is just to develop a concept. Now let's put it in the perspective. If there is a disease which is a very life-threatening disease, bad disease, like breast cancer, for example, right? And if you say that if my tests to catch the breast cancer is less sensitive, very, very low sensitivity or very high specificity, then that means a lot of patients will come to you and get missed out. You will catch the few people with breast cancer. A lot of others who have breast cancer will go home and think they don't have cancer. And then it becomes worse over a few years and they might die. So what happened? You missed out on a bad disease. So in order to catch a bad disease, your test should be highly sensitive. Does that make sense? Breast cancer kind of situation, if a disease is really dangerous, it can kill you, life threatening, you need to have your tests really sensitive. Versus if it's not a life threatening disease, aren't you going to do this all day? Thank you. Okay. So if it is not a life threatening disease, for example, pharyngitis, you do a test called rapid antigen testing. It's a screening test. In that test, I don't care if it's very sensitive or not. If it's low sensitivity as well, we're catching people with pharyngitis and we'll treat them fine. But those who do not get caught, they may still have disease. Big deal, he won't die. Pharyngitis might get treated over time automatically, self-resolving, take care of measures, symptomatic relief, lozenges, etc. Good. Not a life-threatening problem. So I can afford to trade off more specificity in this case than sensitivity to save the cost. To save the cost. So let's put it in perspective now. We said screening tests, we said diagnostic tests. So in ulcers case, we said screening test would be urea breath test. And your confirmative diagnosis comes from endoscopy. Is the only most reliable source. This is a diagnostic test. So that's why we differentiate into two types of tests. So that we can put our screening tests at high sensitivity for life-threatening diseases and low sensitivity for okay diseases.
And this one, sorry, what is ST?
It's a screening test.
Screening test is specificity is more, is higher.
To be honest with you, in urea breath test, our sensitivity is very moderate. Specificity is actually high. Urea breath is approximately 95%. This is sensitivity is 80.
Okay. And in diagnostic one, it should be sensitivity increased in this one?
Actually, in diagnostic test, doesn't matter because they're reliable tests. Diagnostic, 100% you will catch from endoscopy. You're going in the camera inside the stomach.
It means the sensitivity increased.
Yes.
Okay.
100%. It's very high sensitivity. And the reason you don't want to do endoscopy first, cost. Urea breath test is much cheaper. You kept a very moderately sensitive test, 80%. It is a highly specific test. So that I can say, you know what, buddy, I can send more people home. Yeah, you don't have ulcer, go home. And then I'll catch only those which get caught in the disease. And others may be going through later testing and stuff. If symptoms doesn't get resolved in 30 days for something called as uninvestigated dyspepsia, we can get them back after the therapy. We'll say, okay, PPI didn't help. You did omeprazole course for a month. Okay, let's put you through endoscopy now, since it didn't help you. Overall, we calculated this is much cheaper for the whole healthcare system. That's what we did. Does it ring a bell? Does it give an idea of what we're talking about? Now, let's put it in the two examples we discussed otherwise. We spoke about breast cancer and we spoke about rapid antigen test. In breast cancer, mammography is one of the screening tests, right? It is known to be a sensitive test. Good sensitivity.
Mm, that,
Because it's treating life, right?
Hmm?
Because it's treating life.
It's life-threatening disease. And if you are able to have some kind of positive screening out of that, then in the diagnostic test, you will do scanning, which is MRI or etc., just to confirm. So what's happening here? A highly sensitive test. You might overcatch, no problem. A lady might say mammography came maybe positive, but you won't tell them. You will say, okay, we want you to go through diagnostic test first. Is there any problem, doctor? No, don't worry. We'll just want to check more. We just had some little spot to be seeing in the mammography test. We're a little bit concerned. That's all. So you do the diagnostic test and then confirm the results with the patient. Why you did a highly sensitive screening test? Because it's a life-threatening disease. Similarly, you will have pap smear as a screening test. What is pap smear? It's a vaginal test, swabbing. You take the swab sample size of the bacteria. So this is a screening test, a highly sensitive screening test for cervix cancer. And then you will be able to do testing, which will be through proper lab investigation. Da, da, da. Long story short, highly sensitive screening test. Why? It's a life-threatening disease. Now, flip situation. Let's talk about pharyngitis. We have rapid antigen testing for pharyngitis, or we said group A streptococcus, right? Now, if this screening test sensitivity is low, that means we will undercatch. A lot of people will go home and they will still have pharyngitis, no problem. But those people who get caught, we will do throat culture on them. Throat culture is what? It's a diagnostic test. You send the swab, it's costly, long process time. By the time the results come, maybe he's already self-resolved. That's why you don't do throat culture as the first test. Rapid antigen test is screening. So low sensitivity, no problem. But whatever gets caught, you do throat culture and you do diagnosis based on that and do the directed therapy with antibiotics. Did you get the idea of screening tests versus diagnostic tests? Good. Now, for exam point of view, you will have to remember the formula. Just keep in mind that your true guys are on the top only. A and D. True positives, true negatives are on the north, on the numerator of the formulas. Whereas your bottom will have the totals. So sensitivity means all those people who have the disease, out of which how many are we able to catch? The true positives out of all who had the disease. Because high sensitivity means we need to catch. Specificity is opposite, who don't have the disease and their true negatives being eliminated. And on the two bars I created, this half was true negatives and this half were true negatives. So this is easy to eliminate, easy to eliminate. And that's where the trouble lies. And that's what we're working on here.
So, practically speaking, these tests are actually done in all the study processes of a like a disease or something.
No, diagnostic, normal. These are applicable in the real world. I gave you real life examples.
So, how do they count false positives or false negatives?
It's not easy to calculate. It's not. You will never know the patient I sent home actually had the disease or
So
not.
how can you give the number?
Oh, you mean these ones in this study, or you're talking about the breast
No,
cancer
I'm
studies and all?
talking about this whole chart. Like,
Okay, good.
We need to calculate the sensitivity and specificity of something.
Get your idea.
How will we know the number?
Very, very, very hard to know the real false positives and real false negatives. There are some statistical ways to come to know a probability of having how many these, how many that, but they're approximations. So, and that is beyond the scope of us. Only statisticians, epidemiologists, they will sit and figure out ways to calculate those. But the reason why we're doing this is for us to have an idea how these calculations happen. Another thing that will actually complicate if I tell you more on that. This guy is relevant to this guy. This guy is relevant to this guy. That's where these guys come in play. For example, TPFN, TPFN. And the problem is if you guys are having high sensitivity, not TP, sorry, high sensitivity means your false negative should be low. Similarly, if you want high specificity, your false positive should be low. So this is a false negative, false positive situation. If anybody's sensitivity is the problem, false negatives, sensitivity. Yeah, this guy, it should be low, right? We said. Now the problem is the real actual numbers in epidemiologists' real terminologies. He doesn't care about sensitivity or specificity. What he cares about is positive predictive value or negative predictive value. These two numbers. But we, as clinicians, since we don't care about the numbers, we care about the result, we care about the impact on the patient, we care about how many people came with the disease to me and how many people went home with the disease rather than getting caught. So those are clinical implications. Those are my problems as a clinician. So that's where sensitivity specificity is more relevant to me. Once you're able to come to the real grasp of the concept of positive predictive and negative predictive values, it will be relatively easier to get these numbers. That's just to answer your question. But since, as I said, that concept, ignore it. You don't have to know the positive, negative predictive values. Don't worry about the way how you can decipher the gray area. Okay, we're good to go with the formulas. Okay. Now, you don't have to worry about this as well. This is something I don't expect them to ask on EE level. They haven't so far, and probably they wouldn't in future as well. In MCQs, they do, by the way. Sensitivity, you just need the first sentences from here. Sensitivity is the number of those patients who are having disease and who got a positive test result. How sensitive to catch the disease, right? Two positives we're talking about here. Specificity is the number of those patients who are without disease and who got negative result. That is true negatives, right? Makes sense? Like I went to the disease, PUD, I caught from urea breath test. Versus I don't have PUD, I didn't get caught in the test. So these are two different things. We discussed that. Good. So don't worry about what is ruling the disease in, ruling the disease out. As long as your concept is clear on sensitivity, specificity, you'll be good to go. And know the formulas, please. Okay. Now comes some fun stuff. Is everybody comfortable so far with the idea? I know it's boring topic, but but this is what it is. Experimental event rate. I introduced this term before, but I didn't explain more. The idea is in the experimental group, that means those guys were my, this is my experimental group, where I gave them the exposure to smoke, for example, or where I gave them the real medication, not the placebo. I'm experimenting on them, right? These are not my control. Control guys are the ones who got the placebo, or they were not exposed to smoke in the case of exposure, right? So my experimental group is this guy, and my control group is this guy, this group, right? So what is the event in the whole story? What is the outcome? Disease. Those who were supposed to cigarette, they got lung cancer. They got what? Lung cancer. What is that? That is an event. Lung cancer happened to them. Those guys who didn't smoke, they got lung cancer. Yes, some of them got lung cancer. An event happened to them. So anytime a disease or an outcome is figured out, that is the event. So out of all these are the people who got the disease or have the event. So out of my smokers, these are the guys who got the disease. Out of my non-smokers, these are the guys who got the disease. So event rate is of these people out of smokers and these people out of non-smokers. Does that make sense? So if, for example, this was 1 and this was 2, total, for example, was 3, this was 2 plus 2, for example, total was 4, then I will say 2 divided by 4 is my control event rate, 1 divided by 3 is my experimental event rate. Let's do this again. Experimental event rate is a measure of how often a particular statistical event, like a disease, occurs within the experimental group. So out of this group, guys, we said exposure happened, not happened, disease happened, disease didn't happen. Now we're saying these are the event guys. These guys got the event, the disease. So what were my experimental ones? These ones were experimental ones who were exposed to the smoke. So the total, whatever number is here, A divided by, let's say, E, is your
A plus B.
A plus B. A divided by A plus B. Similarly, control guys are the bottom ones here. So control guys, yeah, bottom ones. So these control guys, and the event happened is this. C divided by C plus D, A divided by A plus B.
Why we didn't say D is divided by C plus D?
Very good. Let me explain that again. Those guys who, you're saying why didn't we take these guys? The question is, they don't have the disease. They don't have the event. See, does not have the disease. We're looking at the guys who have the disease. So these are the people who have the event.
Please, can you go to this formula? You said that memorize this formula for specificity and sensitivity.
That is different.
Oh, this one.
Yes.
Yeah.
That is different. And I said there is an overlap of the formula, right? A divided by A plus C. No, not that one. Sorry. This is the formula which we were talking about. Positive predictive value. A divided by A plus B. Doesn't this look like experimental event rate?
Yeah.
And then we were doing this. I said there will be an overlap, but don't worry about the overlap. That doesn't mean they're the same thing. They're very different. Formula is similar. Numbers might be similar. No problem. And as I said, you don't have to worry about this. So just keep in mind, experimental event rate means what? This is my experimental group. This is the event rate. A divided by A plus B is what? Experimental event rate. C divided by C plus D is what? Control event rate.
And this one we made D divided by D plus B.
This is different. Again, please don't mix the sensitivity specificity concepts with them. But if you want to just do for the sake of memorizing your formulas, maybe I can help you very quickly. Sensitivity means
No, I got the idea.
Got the idea?
Yeah, no need to.
Okay, good.
I just confused between this one and the new one. Experimental.
Sure. Okay, good. So as long as you got the idea of these guys, you need to know how you're picking this information up. Now, what will happen is in the exam, this will be the first thing which you need to pick out from your question. Whenever they ask you an epidemiological problem, a case study, or whatever, the first thing you want to extract are the event rates so that you can do further calculations. The further calculations, all of them are based on these two. If these numbers are wrong, question is gone. You got everything wrong. And they're smart enough to put those answers as other options. Whichever wrong answers you will get, you'll say, yes, it's there. And you select and move forward because you got the wrong one based on these numbers, right? So how do you want to pull them out correctly? They will say, for example, in a question, out of those people who had ulcer, 5 of them, no, this is a bad example. Let's take lung cancer again. Out of 10 people who had lung cancer, 5 of them were the ones who were smoking. Out of those who didn't have lung cancer, 6 of them were this, da da da. So what you're saying, immediately you have to catch control versus this, experimental. From the vision of the question, you have to be able to be trained enough to pick up these numbers immediately. Either you make a 2 by 2 table, which I highly recommend that you do. If you don't have time, you should immediately be able to say, okay, 5 people out of 10 got in the experimental group that got the drug, 5 divided by 10. 6 people out of 10 were the guys who were taking placebo, 6 out of 10. Immediately you came to EER and CER. That is where the training level has to be at. You should be very rapid. And if you're able to come to that, that's not the end of the question. Questions will be usually way longer than this. The reason why I'm saying it, you won't have 5 minutes per MCQ. You will need to go fast, really fast. You will have minute, 70 seconds, 72 seconds per question. You don't have more time than that. Just a minute or slightly over a minute. So if you take 2 minutes just to calculate these, what about the rest of the question? So that's what I'm saying. This is the easiest step. We need to do more training to pull this information out. And I have 5 examples today to practice as well, so we'll be good. Okay. Now, what was I going to say? There was something important I wanted to mention. It just flipped from my mind. Anyway, so new formulas. Oh yeah, I was going to tell you what type of question they might be probably asking. So sometimes they will say this is a case study, and they gave you 4 options. They will say experimental group. There were people who were given drug. These many people were able to get the treatment or solution out of these many, or got the disease out of the, and placebo was given to these people. You got the numbers. Then, which of the following is true about this study? One of them will be, for example, number needed to treat. They're asking about this number. One of them might be RRR is equal to a number. They will sometimes say ARR is equal to this number. So what they're doing is they're essentially asking you 4 calculations in one question. 4 different calculations in one question. So that's where these are, even though long, but believe me, once you're good on these, they are easy marks. These calculations are easy marks. And Route is going to come and do calculations with you, but I want to lay a little bit of an idea to you. Approximately 22, still 22, still 22 or 24. 22 calculations approximately are going to be there in your exam. Combination of different types. Some might be pharmaceutical, some might be management related, some might be financial calculations, some might be the
Dose
the
calculation.
the dilution concentration ones. What's the allegation method? I was saying algorithm. It was, so allegation method. Some might be just epidemiological studies. At least 2 questions, for example. So these are easy ones. Out of 22, 4 or 3 or 5 maybe pharmacokinetics ones. Out of pharmacokinetics, 2 or 3 might be very bad and very tough, like flight all over your head, and you'd be like, don't waste time on this. So -3, you can still score really well on calculations. If you are just someone where you missed two calculations, no problem. You can still score good on calculations. 17 out of 22, 16 out of 22, I'll take it any day.
And they are easier in exams.
So the easier ones, focus on them more. Get them as your strength. Don't keep looking at your weakness. But Mohsin, I don't know kinetic. Okay, don't worry. Do you know epidem one? No. Then what's the point? You need to know the easy ones first. Know them really well. Make them your strength. That the moment you get them a mark, 1 point is 100% confirmed. Then those ones which are difficult ones, we'll figure that out later. No problem. Okay.
But the problem is they put all the calculations in the beginning and they put the epidemic calculations at the end, where when you don't have the time to do all this, like
No problem. So that's where you'll have to probably pick and choose. You'll have to probably go according to the format. The times when I was giving EE, the calculations are right in the middle of the exam. In the beginning, they were asking single line questions. Calculations came in the middle, and at the end, starting question number 85 or something, they used to have case studies all the way till 150. Case studies, case studies, case studies. So first day of the exam, I had a problem because when I was doing case studies, like a whole page to read and one little question at the end. So I had a problem of time at the end. I couldn't read and I couldn't comprehend because of panic of time.
that's
So
what happened to me as well.
right. So what I did, second day when I went, I started question paper exactly opposite. I started question number 150, 149, 14. I did all the case studies first. It took half of my time, no problem. But then now I'm left with are one-liners, single
You
questions
need
that
to
they're
read.
easier to answer.
You need to read these cases. Sometimes some people say is this a big case and just the question, no relation with the different.
Many times it might be relevant too. For example, someone might have an allergy in there somewhere. Someone might have some kind of mention. So you need to be able to have quick reading skills, something called as skim reading. You need to be able to quickly pick out some numbers. Blood pressure is here. Go for blood sugar is here. Oh, allergy is here. You need to go forward.
Like eyelids test.
Yes.
We need to read again.
So if you're able to do those, and then calculations. Well, here's the thing. Calculations have been my negative as well. Very bad weakness it was. All of us are in medicine because we didn't like maths. Simple, right? Otherwise, we would have been engineer, whatever else, right? So if your math is weak, you're here. And all of you, all the students who come to me and says, Mohsin, math, I don't know calculations. 100%, not 99, 100%. Actually, I'm wrong. One exception is there, that is Pashupati Raut. He was my student of OSCE, and I was like, calculations, and he's like, no, they're good. It's like, really? You're the first person who says that. And his calculation, he knows his mind is very mathematical. I said, can you help me? Can you teach my students maths? So now he's helping me. Raut is the guy who's coming. He was my student before. So the point is, calculation is a weakness. I understand and appreciate that. So we need to find a way to make it our strength. And the best way to do is to go by our own strength. Easy ones, make them your strength. Pharmaceutical calculations, some of them are very easy. Some dosage calculations would be easy. Financial ones, I promise we'll do them well. You'll be good. So do some good ones, which are your strengths. The moment you open the paper, search for the calculations. Where are they? Show me. I want the calculations. The moment you get the calculations, okay, start. If you find a question which is out of those 3 or 4 bad ones, don't panic. Skip. Go to the next one. Where is my epidemiology? Okay, I'll answer that. Management one, I'll answer that. Immediately done with them, the bad ones, then you come to it later. Don't let that bring you down. Mark them, do whatever, leave them for later. Go back to your case studies, finish your case studies because there's reading involved, comprehension involved. Keep looking at the time, calculate time. We'll do the timing practice in mock exams as well. I've got at least more than 10 ready now, exams, which will be helpful so that you can practice your timing when you're doing at home or in the class. So keep looking at the time because they won't let you have watched. There will be a big clock on the wall. So they'll be letting you only watch from there. You cannot put a stopwatch. You cannot do anything like that. So you'll have to calculate based on a normal arm clock. So you need to have practice on that. We'll do the training in the mock exams in December. So once you're doing that, You know how much time left. So you will be able to pace up, slow down accordingly. So you can actually, and usually it'll be pace up. So you'll do the case studies, finish the case studies, then say the individual questions, because individual questions are easier. Which of the following is the type of an RNA and da da da? Okay, this one. Which of the following is da da da? Okay, this one. Easier to do, rather than a patient had this and that and that. Long story short.
My problem was in the first exam, valuation exam. I did the solution in the question. At the end, I found out, oh, there's sheet I have to answer it. That
Oh,
was not what killed me.
oh man, that is fatal mistake.
There was no time.
One student made a mistake by answering one question ahead on the bubble sheet. Question number 10's answer was 11 on the sheet. And she said just 2 minutes before the question paper was, the time was finishing, when she came to 140 and 150, she was like, it doesn't match. One question is extra. Then she saw and she was like, she's one question ahead. And then she started rubbing, and she tore the paper. And she was like, can I get another answer sheet? They're like, time up. Gone. So she lost the entire day. She didn't even bother going to the next day. She said, no, I can't. I tried convincing her that still go. It will be experience. You will learn. What if you get saved? What if this 150 might help? No, I did everything. She didn't go to the second day of the exam. So it can happen. But the thing is, I blame myself for advising her to pick and choose. Because if I would have told her start from question number 1 and go all the way, it's easier. You don't miss out. So this is the downfall of picking choosing. If you start with calculations first, case studies second, you have to be very, very particular about picking the correct number. How I would do, the moment I pick up a question from the exam paper, question number 120, question number 120, question number 120, I will search question. I'll be talking to myself in my mind. 120 answer A, 120 answer A. Okay, there you go. And I will tell you again and again and again. Every time you fill a bubble, you can go back and check your question again. Okay? So try to do that.
I did that 5 times for every question.
Yes.
Then put it
And
in
if you
the
miss, or if you leave a question for later, do 2 things. Mark it on that circle, and upper corner of the page, mark it that I have a missing question on this paper. Because at the end you're flipping, you won't be able to find the question. On the top of the paper, you have a mark that this page has an empty question left. So some basic tactics we'll do few days before the exam. Again, we'll revise this, but it's just to give you an idea. Now, calculations back again. Let's make strength. These are easy ones. Practice them more. ARR is what? Absolute risk reduction. Risk reduction means in the experimental event versus case versus controls. Disease event, I want to decrease the risk of disease. So what's the difference between I give some medication to the others I didn't? Then what's the difference of disease between them? Who got more disease? That's the difference. Risk reduction, I want to check. Risk reduction of the disease, which decreases the disease in which population more. So that is my risk reduction generally. Then there are 2 types of risk reductions: relative risk and absolute risk. Now, relative risk means relative to, how to explain it? In simple words, relative is supposed to be based on how no, that doesn't make sense either. Your experimental group versus control group, the difference of disease rate is absolute difference. Okay. Now, relative would be, how to put relative, the difference versus the difference. That means ARR is CER minus EER divided by CER. Yes, that's correct. That is control. Okay, now good. So we're trying to use one of them as a base, as a gold standard, and then we're comparing the other one against it as a relative.
Like bioavailability with IV.
Kind of, but this is relative to this. So we're saying that it's not absolute, it's relative to my gold standard. You know what I'm trying to say? Let's put it in numbers and it will be helpful. ARR is talking, what was CER? How do you calculate CER formula?
C plus C. C upon
D
C plus
upon
D.
C.
C divided by C plus D,
This one is called
right? Whereas your experimental is what?
A upon A
divided by A plus B.
A plus B. This divided by total, this divided by total. These are my experimental event rate, control event rate. So you will calculate your control event rate. For example, it was zero point 0.5 and you did 0.4 and you found out your ARR is this one. There'll be a situation, there will be a situation where your EER might be higher than CER. Then what will happen? 0.4 minus 0.5. You might get minus 0.1. So the idea is when you have the word absolute, you put it in something called as straight brackets. Straight brackets. That means negative sign is irrelevant, right? So whatever the sign might be, the answer is going to be 0.1. Whether CER is higher or EER is higher doesn't matter. So in absolute risk reduction, the real formula is straight bracket CER minus EER straight bracket. And then whatever the sign of the answer might be, it's neutralized. Okay? Relative risk reduction, on the other hand, what was ARR?
Absolute risk reduction.
Formula is
CER minus EER.
good. Can I not write this like this as well? That's the same thing, right? And this one is of course in absolute bracket. So I just put the ARR formula here, right?
Why was CER though?
CER is this one. We're
Sorry,
looking at the
sorry.
RR
You
formula.
this side.
I just replaced ARR
Okay,
with the
okay,
formula, right?
okay.
So this is the difference between ARR and relative risk reduction, because you have a relative as your gold standard against which you're checking the absolute risk reduction. Tells you how much the treatment reduced the risk of bad outcomes relative to the control group who did not have the treatment. Formula is this one. And again, as I said, if your CER or EER is wrong, everything else is wrong. You see? So you need to make sure these are correctly diagnosed, correctly picked up. Then a number is NNT, number needed to treat. That means how many people I will dose with this medication until I'm able to eradicate one disease. One person is totally treated. How many people I will dose with this medication until I reach to at least one outcome of result, a good outcome, for example, right? So number needed to treat. How many number I need until I get to treatment? And it is the number of patients that must be treated to prevent one adverse outcome, for example, or prevent one heart failure, for example, one heart attack, one myocardial infarction. That if we're giving aspirin, and the number is crazy high, I think it's 1800 and 1800 and something is the NNT of aspirin, 81 milligrams cardioprotective dose for preventing cardiovascular disease. So you gave dose of aspirin 81 milligrams to 1800 people, then one heart attack is prevented. Right? So that's what it is.
Why all doctors write specific or
Because we're able to save one life. Aspirin
and
is
there
cheap.
is another way. There is no another way for this patient.
Other medications are there too. Aspirin is relatively cheap, very, very cheap. It's not cost at all. And
But
it's,
this is not effective. What I will do is cheap and not save the patient.
they save
What I'm
one.
doing.
It saved one after 1800. It could be you, that one
Yeah.
person.
No.
Then it's worth it,
I'm
right?
not try aspirin. I'll go for more higher price. I want my life.
Okay. So number needed to treat. Okay, got the idea? You might sometimes see a thing called number needed to harm. It's an exactly opposite idea. Formula is exactly the same, but you don't have to worry about that. I hope they will not ask you this. If they do, NNH is just like NNT, same formula. 1 divided by ARR. Then we have a thing called relative risk. Now there are two concepts, RR and OR. Now you're going to differentiate the two. Are we taking lunch break sometime?
Hopefully.
I'm starving.
Me too.
I can't stop eyeing my carbohydrates. They're lying there and I'm just looking at them like lollipop.
I think we should take one. Within 5 minutes.
Or maybe now.
Yeah, well, just left with 2 slides only.
Okay. So, relative risk of a bad outcome in a group given intervention is a proportional measure estimating the size of the effect of a treatment compared with other interventions and no treatment at all. Who understood this? Nobody?
I'm waiting.
Me neither. Okay, so let's make a concept. By the way, what did we say? You use RR in which type of study? We said relative risk and odds ratio. You use which one in which case control or cohort?
Relative risk in
Yeah,
control.
in the future study.
Cohort.
Yes. Cohort study.
And odds ratio in
Case control.
case control. Thank you. So odds ratio is in case
No.
control.
No.
Yes. Odds ratio is in case control. Relative risk is in cohort. Okay. So now we're talking about those two numbers here. Oh, what a mess
Oh,
I've
yeah.
created. Okay, so relative risk of a bad outcome. Okay, so the formula firstly is EER over CER. That means your experimental event divided by the control event. So bad outcome in a group given intervention, this is one group who was given intervention is the experimental group, is proportional measure estimating the size of the effect compared with other interventions or no treatment at all. These are what? Control group. So we're saying what is relative risk? Is the relative risk of the bad event. Proportion of E to C, experimental to control, proportional measure estimating the size of the effect of treatment. So how much is the relative difference between the experimental versus the control in order for me to decide whether this therapy is going to be relevant or not in case control studies? No, in cohort studies, sorry. Let's eat food.
Yes, I prefer because
I didn't get the difference between relative risk and absolute.
Okay, Ibrahim, Omega, are you guys there? Because we're going to start now. Okay, good.
Yeah, I'm here.
Okay. Okay, so the two terms we were going to define now were the RR, relative risk, and the odds ratio. Relative risk basically generally means the two groups, the experimental and the control group we're talking about, both of them have their own risk factors. Control group has a risk of getting the event, heart attack for example. Experimental group has its own risk of getting an event, the heart attack for example. We're just comparing the proportion between the two guys having their own risk factors. Risk factor of heart attack in experimental group after using that medication, risk factor of having heart attack in control group without using that medication. That is just relative risk. This is the formula and you should be good to go.
So we are comparing which one is more prone to the risk.
Yes. The idea is which one is not prone is not the idea. You're just comparing both of them having their own particular risk factors and the proportions. More or less is a relative here. The idea is this much is there, this much is here, total.
So compare to each other, which one has more?
Goal is not that. Goal was in relative risk reduction was the goal. Which one is more or less? How much reduction of a goal is? Here the idea is the proportion. Where is the proportion? And then you interpret RR in so many ways. Interpretation is different. Right now what you're doing is you're interpreting it already. You're saying that which one is more. That's not the interpretation we want to come out of it. We're just going to say RR may be significant, maybe not, maybe more, maybe less, whatever is there, that's the interpretation part of it. Odds ratio, though, on the other hand, is an association between a risk factor and a disease by comparing the likelihood of disease under two circumstances. Two possible circumstances. Now, odds ratio is basically which one? We said A, B, C, D. Now you're going to be cross multiplying the two numbers. AD divided by BC.
Okay. Because the A was true positive and D was true negative. So they have the
So you're talking about not real negatives, actually trues divided by falses.
Yes.
All the trues divided by falses. So anyways, the idea of odd versus idea of the probability idea, it's a whole story in stats, which we'll be doing biostats someday. And even there as well, I will try to skip that through because it will take me 2 to 3 hours to explain the whole idea of probability, the p-values, the difference between probability and odds. It's a long story. But for you, for exam point of view at this time, we can give you what we can. And if you guys need, anybody is interested, by God, I have no problem sitting with you 2-3 hours and explaining the whole idea of odds versus the probability. So I recommend not getting there yet. No need to go into that. Okay? Because this is itself good enough.
And that's what I like to hear.
So let's quickly revise the formulas we have discussed so far. So we've said, what is EER?
A upon A plus B.
Oh, do you need the formula?
Do we agree? Yeah.
Yeah. A divided by B by A plus B.
A plus B. What is CER?
C upon
C upon C plus D.
C by C plus D. These guys? Everybody with me there?
Yes.
What is ARR?
CER minus EER over CER. No.
No.
Minus A.
CER minus EER.
Yes. That was added.
One not here.
That's the formula?
Yes. AR.
What is NNT?
1
One
upon
upon
ARR.
What is RRR?
ARR
minus EER.
ARR upon
ARR divided in CER.
Good. Or you can also say it as
CER minus EER divided by CER. And what is this lines?
straight bracket. Then what about RR?
EER divided by
CER.
minus CER.
No, divided.
Divide by minus or
Divided.
divide by
CER.
Because minus was ARR. This is CER divided by CER. What about odds ratio formula?
ADBC.
yes. ADBC.
We know the formulas now? Let's apply. So we will do this question. Don't look at the answers in your book or anywhere. You calculate and finish the question first, then we'll see if you have any problem, whatnot, etc. Okay, so you use the formula ARR divided by CER.
CER, yeah.
What was your ARR?
A
0.01. Sorry.
A
Is
0.003.
0.0.
Before that, what is your CER?
My CER comes out 0.317.
0.31.
20 divided by
20 plus
63?
43.
Yeah.
Yeah.
Yes.
33. 63, yeah.
63.
0.31.
317.
31746.
And EER is
0.36.
102.
32.
0. Point what?
313.
6.
313. Okay, what was the you did 30 2 divided by
102.
102.
102. Then why is your number different, Ibrahim?
It's 0.36 and the other 0.31. Have to be. I didn't put the third number. We have to use 3 numbers or 2. I use just 2 numbers.
See, before that, doesn't matter if you use 2 or 3. 32 divided by 102 is 0.313. Why are you saying 36?
The second one is, yeah, it's 36. 32. I divide this 37. Oh, sorry, I divide 37. 32. 37. Oh yeah. Sorry, I divide maybe 37 because that's
So
the
you
reason.
got two numbers now, CER and EER. ARR was the formula was
CER minus ER.
32.
and the answer was
I got 0.004.
I got that too, but when I took 5 numbers together, then I got the right answer as given 0.003. So I don't know why.
0.004. Okay.
I get 0.003. And this is also right. When
Right.
you
And you
divide
took more numbers than 3 digits.
this is the 5 in the fourth place, but I didn't use it in the
Yep,
next
yep,
question.
yep. We'll talk about it. Don't worry.
Okay.
So
I put the 37 majority in bad. So against 32, I put 37. That's the reason.
yeah, okay, no problem. This was your wrong answer. Anyways, now the point is, first things first, there are a few steps which you're gonna do. Whatever numbers you calculate, you need at least 3 guys after the decimal. You can take 4 as well, but at least 3 guys. For example, 0.003, you need at least 3 people after the decimal. Reason? Once you multiply that with percentage, you will get at least, for example, you will have decimal coming in here now.
Yeah.
Right? So that's the idea. If, for example, there is 1.333 and you bring it to percentage, you need at least 133.2. You need at least one guy after the decimal. So in order to have this guy, you need to take at least till the third one, at least. If you take fourth as well, no problem, which is okay. Here's the problem. In the question, in the exam, you have options. Out of your 4 options, if you're able to eliminate 3 and you're calculating this guy, you say, okay, I think this is the correct answer. If you're doing that, if you come to one option, then stick to your same idea of 3 digits. So start with 3, go to the formula of 3 after this decimal, keep 3 guys included. And if you're able to come to your conclusion answer, you're good to go. But if you somehow say, this is eliminated, but I don't know about these 2 guys, both of them seems okay to me, then get another decimal. Then you can spend more time on it. But otherwise, usually you'll be able to come to a good conclusion immediately with 3 decimals. Okay. Then, in our case, we did more decimals and we got this number, but it's okay. We got 0.004 in our calculation here, which is also correct. Okay. Then,
This is give me another answer in
different answers.
NT.
no problem.
Yes, me too.
It's totally different.
Yes, I know there's a huge range difference.
Yeah.
Yes.
Yeah.
Either it will go all the way between, yes. So the idea with N and T is the more further you go for one number, you keep calculating further and further and further into more, the number will keep getting different and different and different. No problem. Here's the idea.
Okay, no problem. But
If
in
you
the exam, there
no
is
no,
a
you
lot of
take 3 decimals and you'll get 2 good answers.
and I'm sorry, I want to understand. But this one, if I took only the three, it's the right answer. And if I make this one for the nearest number, like you did it for, it's give another number. So
Yes,
it's
because there'll be times in your calculations you might get a lot of numbers.
Yes.
That doesn't mean you will have to take them all. So you will take the 3 guys after the decimal, work with those numbers, whatever number you get should be in that idea. And the reason why I say that is because Canadian guidelines are about 3 numbers.
Why
You
don't,
didn't get
to be
my
on the
point.
safe side, we use all the numbers?
If we do, your numbers would probably be different than the exam answers. The more closer you want to be to your exam answers, 3 decimals are the way to go. Guys, I have 3 points after decimal. My answers may or may not be 3 based, big deal. But if you've done 3 decimals and your answers came, that will be highly likely in your list. As I said, it will be there. Canadian guidelines recommend you.
If both of them is they're in the exam, what I should do? Like this one, and this is your answer, three or so on like that. What
Stick to the answer with 3. I repeat,
this
Canadian.
one was three. I just make this one four instead of three. This is the nearest number. You know what?
I agree. This is the nearest number too, dear, to think of it. Does that mean you'll sit all day with the 20 numbers? No. The point is, I repeat, Canadian guidelines are for 3 guys after decimal. Stick to it. Whatever numbers you get, tick mark in your question. Simple. Does that make sense? 3 after the decimal is Canadian guideline. You're giving Canadian exam, right? So you have to follow their guideline. Even though I know it's not the closest to the real answer, it may not be, it's irrelevant. What's more relevant is what they recommend you to do. They recommend you to take 3 guys after the decimal. Stick to that. Whatever answer you get will be in the list. Okay. So we calculated ARR already. Then what is your RRR?
0.01.
0.01 plus minus.
Yeah.
Okay. You got the same? Everybody got the same? Okay. 0.012. Okay. Yeah, which is fine. Which she got the third one too, which is okay. Third one is acceptable. Then we have odds ratio. This should be easy.
0.98.
Oh,
No?
okay. No. It's
0.98?
Yeah.
0.98 is correct. Yeah. Now NNT would be different because your ARR was different, right?
Yeah.
I have 250.
Yeah, so the range would be between this guy to this. Is that correct?
Mhm.
Everybody,
No, for me, I did not get it, the last one. And
you didn't calculate that one? Okay.
It's 1 upon 8.
So 1 divided by ARR is NNT, right? The formula?
Mhm.
Yeah. So 1 divided by your number of ARR was 0.003, or some of you might have done 0.004. So from either way, your answer would be different. So this is the range, 250 to 333. Okay. Some of you would get in this range, some of you will get in this range. No problem. But if you took 3 decimals, whatever the answer is, that's more closer to what Canadian guys would want you to do. Okay. Next question. And let's try to do it a little more quickly to practice for the exam. Maybe they may not even give you the table. Now we have a table, it's easier. If it's written, then you have to make a table yourself. We need to learn how to make a table also eventually. Okay? So let's go quickly.
What we are calculating?
The sensitivity and specificity of this new tool.
I did it already with last question.
Good. Is this tool likely to lead to significant unnecessary medicating? So please go through this test. So sensitivity formula is A, B, C. Is A over
A plus
A
B.
plus C.
and specificity is
D
D
upon
D
D
upon
plus
D plus
C.
D.
D plus B. So are these your answers?
Yes.
Good. Now let's say sensitivity is what the number was,
The sensitivity is 0.7.
0.7. Oh,
and specificity was
1.
1. Now the interpretation is important. Now they might ask you interpretively. They might ask you the question like this: Is this too likely? Is this too likely to lead to significant unnecessary medicating?
It means catch more,
Specificity is
more
so no.
sensitivity.
You mean in order to catch more would be higher sensitivity? Yeah. In this case, sensitivity is lower, specificity is more, right?
Specificity is more.
Specificity is more. So
Yeah,
answer is
yeah. No.
no. That means we're not catching more. We're under catching.
Yes.
Be more specific.
In more specific, you're under
Yeah.
catching. You're
Yeah.
catching lesser than.
Less.
Yeah.
Yeah. So the answer will be you're not medicating unnecessary, right? No, this tool is not likely to lead to significant unnecessary medicating as it has a high specificity. A high specificity means that if the patient tests positive, they are likely to actually have the fungal infection, in which case treatment is warranted. Only if they make it through this difficult testing, then they. Okay. Does that make sense? Easy?
Yeah.
Good. Now let's try another question.
Oh, we should make it.
So, first thing what you have to do is, as I said, if you're able to pull out the chart, they make the contingency table, 2 by 2 table, which is ideal. But if not, in order to make it quick, you have to be able to have a bird's view to catch it immediately. That means new drug was tried on these people. So immediately you know the drug is what? Experiment. The total number of people are these. From new drug, 28 got side effect, event rate, and 7 from the placebo. Placebo guys were these. So this should be my control guys. These should be my experimental guys. Does that make sense?
Yes.
Even without making a chart, you can pull this out, right?
Yes.
But if you're willing to make a chart, that's even better.
I prefer that go with a chart if that
Sure.
I think will be make more easiest if you
In the
could do it.
At least in the beginning, when you're practicing more, make the chart, get practicing done. The more examples you practice again and again and again, then you'll be able to come to a point where you'll automatically be able to take the numbers, because in exam time would be short. But to be safe, you have to, if you want to, you can make the chart depending on how good you are with the idea. Everybody did good till this step, or any issue? We fine? So this is what CER. The numbers you found were what? Any total numbers you got? EER was what?
We didn't
We
go for
didn't go
EER,
for this one.
It's just odd ratio.
Oh, sorry, my bad. Haha.
Oh, dear.
It's me. I didn't even see you're looking for only odds ratio. All right. Okay, now I see what the problem happened. You anyways had to make a chart to do that. Okay, good. So my chart is telling me what? Disease, no disease, exposure, no, drug, no drug. So this is placebo, this is drug,
Oh, it's the same. We did it.
Yeah.
We
and
did it
side
this way.
effect and no side
No
effect.
side effect.
Then we said how many people with the side effect and the disease?
And the disease is 28.
Sorry, it's 28 people got side effects
Yeah.
out of the
5 to 6.
5 to 6.5.
out of the drug guys. 28, 5 to 6, 5 is the grand total.
It's 5 to 3.7.
5 to 3, 7.
Why you put it in placebo?
Not here.
It should be in side, no side. This one.
Sorry, one
Side
sec.
effect.
What was the total number of guys the drug tried on? Okay, yeah, sorry, my bad. all of them got the drug. 5 to 6. Well, let's make it from the beginning. I'm finding it hard here. Okay. So we said out of my people 5265 who all got the drug,
Yeah.
my 28 people got the side
Side
effect.
effect.
Side effect is the event,
Yeah.
the outcome that is,
Mhm.
they got side effect. 5237 you said is the leftover amount. Then all the people who got placebo, total is 5000. Out of them, 7 got the side effect. Side effect, yes, in 7. What is my other number?
4993.
4993. And then you cross multiply and get the answer. What was that?
I got 0.262, but the answer is 3.8.
3.81.
262.
3.8 is come when we didn't deduct anything. Just take as it is.
Did I?
No.
Like multiply, I did this. Like 20.
I get
Sorry, Mohsin, can you repeat again the chart?
I also got 3.8. If we don't.
3.81.
We just take.
Okay. Now, as we said earlier, this is the flip model. 5237 multiplies by 7. 28 multiplied by 4993. Sorry, Ibrahim, you had a question. You were saying what? Read the chart. I will do that. Just one.
Yeah.
By the way, this chart is not correct. The one we made is more correct. Okay.
Yeah,
Okay.
And the answer we are getting 0.262.
28 into 4993 divided by I don't know how my phone works. Is this how it works? Yeah, there you go. Okay. Where's the other one? Yeah, 7 into 5237. Close the bracket. I got 3.81.
Yes, 3.81. If we got cross multiply, my answer is same too.
Then what's the confusion?
I don't know what they did.
If you multiply these together, then answer has to be 0.381.
Yes.
Okay, let's help Ibrahim first and then we'll come back and do further calculation again. Okay, so Ibrahim, to begin with, we're saying side effect is the outcome on the north and then the drug, which is the exposure on the left. Drug given, placebo given, that is negative for the drug. Side effects happened, no side effect happened. This is a total and this is a grand total. Okay, so now you're saying the new drug was tried on 5265 patients. So all the guys who got the drugs, the total number is 5265. And placebo was on total 5000 people. Placebo, 5000 people. Now then we said from the new drug, out of the new drug people, 28 got the side effect positive on 28 people. And 7 got side effect from the placebo. Placebo, 7 people plus side effect is there. Then this minus 28 is the number which was 5237, am I right?
Yes.
And then 5000 minus 7 would be 4993. So this 5000 minus 7, you'll calculate this number. 5265 minus 28, you'll find this number, right? Because this plus this is the total. 7 plus this is the total. That's the idea. Then you came to your 2 by 2 table already now. And now you can cross multiply for odds ratio number and you can get the answer 3.81.
So always when we have to make the table, we will see even if they don't give it in the question that these are the people who do not get the side effect. We have to make the chart in the two groups, like the one which gets the side effect and one which do not get the outcome. So the ones with the outcome and ones with not the outcome.
Yes,
Always.
that's right.
So they asked in this kind of question or the first they give the chart. So all
Either.
in the question usually.
Either possibility is there. Either that one can come or either this can come. Both possibilities are there. So you need to be able to spend some time to know how to make the chart. That's the reason we spent some time in the beginning defining ABCD, true positive, true negative, just to have an idea how to make the chart. Outcome comes on the top, exposure comes on the left, etc. Okay? Anybody having any confusion? I'm going to give you the new chart as well, and we'll change this one. This one goes out because this is based on the flip model. And remember, in the beginning, we specifically said that we're not going to use a flip model. We're going to use what? We're going to use this model. This is what you have to just ingrain in your mind. For this, for MCQs, for future exam as well. Okay. So now let's talk about another calculation. Okay, so here's the idea. The reason why this is different is to make sure that we know our material well, we know where it goes where, rather than just picking up any chart. And if you remember the upper left square and the lower right square, then you're gonna lose. So that's why you need to read to understand what they're talking about. Clinical diagnosed depression is the outcome. It stays on the north, whereas scale or test or exposure stays on the left. This is correct. What is wrong is this is negative, this is positive. Instead of what we studied was so far this one, right? And this is no and this is yes. What we studied was positive and negative, right? So how do you flip the chart now?
I took 17, then 20, then 126, and 6.
So firstly, these are true positives. 17 yes and a yes. No and a no are true negatives. Does that make sense? So 17, 126. That's according to your chart in your mind?
Not me. I didn't do this.
No, this is the original one which we remember. This was supposed to be true negatives, right? This was supposed to be true positives, right?
Yeah.
Then all the positives are on the north, we said. So what are the other positives you have here from this? What is false positive from this chart?
20.
False positive means what? That they got tested positive,
But they were
But
not
then
positive.
but they were not positive. So there's a yes on this guy, but they were actually not diagnosed with depression, right? So these guys, 20 people will come here. This is actually what? These are false positives. Yes means positive, positive. No means negative, negative, which is false negative. This one.
No, these are false negatives. So, 20,
The
126,
126,
no,
26
20
should
is
be
false
on the-
positive. Yeah, 20
Yeah,
is false
6 should come here.
Yes.
Now this is in the format of the way you know it. Is that right?
But I put 126 above.
I put like 6 in the top over there and 17 down, and in the right I put 126 and the bottom 20.
It will give us the same.
That's what I put.
Will give the same answer, but the sensitivity in
Exactly. Now the idea is,
Yeah.
if you go for the original chart and make your own calculations, your sensitivity will be specificity and your specificity will be sensitivity, and your clinical conclusion would be wrong. My question would be then, is this a sensitive test to catch depression? Is it a good test to catch depression or not?
I have idea. Instead of I waste of time, I'll make my chart then convert the answers. Is it okay?
No.
No.
You never know in what format it comes. If you're able to bring it in your format, good. First question you need to be able to is catch that there is a problem. That there is a problem. That means you cannot simply just close your eyes and pick whatever information is in the squares and make your calculations. And that's the only thing I want to bring your attention to here. Okay. Sensitivity is supposed to be 0.739 and specificity is supposed to be 0.863. Those who followed the same chart, they flipped the answer.
But there is another question related, will be wrong later.
That is. Oh yes. If they ask further things and say, firstly, you lost marks here. Secondly, if they ask you which of the fall is this test good to diagnose depression? If they ask you that question, is it sensitive to diagnose depression? And you will say, no,
Mhm.
sensitivity is low. But if you instead ended up saying yes, because your sensitivity came higher, because you flipped the answer, then you lost all the 3 marks. Right? Anyways, again, my emphasis is out of all the 5 examples, first thing, you need to know how to make the 2 by 2 table. Very, very well. You need to know the places of your outcome. You need to know place of your exposure, or in case of drug, the exposure or the exposure or the
Test.
test. Thank you. The test. So either test or exposure, all of them are going to be this side. Positive, negative, positive, negative. You need to know these are true positives. You need to know these are true negatives. You need to know these are false positives. You need to know these are false negatives. Because positive means uh, positive, positive. Negative means negative, negative. Okay. Then you need to know what totals, how you can calculate totals. This plus this is this. This plus this is this. FNTN is this. TPFP is this. These are just totals. Just normal 2 by 2 table. Now, once you know how to make your general table, then you can put information. Then you need to know, number 2, your formulas. Well, you need to know what is CER, EER, RRR, ARR, 1 by, that is NNT. You need to know RR, you need to know OR. These are some basic formulas you really need to memorize. They may not give you those formulas in the exam.
Even though I got the answers right, but the thing is, I had nothing in my mind about true positive or false positive. Nothing about that. I just considered yes as experiment and no as like no placebo, no placebo. So I took it that way. But answers were correct. But will it help me with further questions to?
Yeah, sure. As long as you're able to get to the right numbers, you know which number is doing what, that's good. But ideally, if you're able to come to the idea of true positive, true negative, that will be able to help you. If they make the question more complicated, then you can easily come to the conclusion. So anyways, first thing is you need to know your table making. Please, formulas are important. Then third thing, whenever you're taking, at least take the 3 decimal, the third decimal maximum, minimum you need this one. That is usual Canadian guideline. And then what? Then just make the maths correct, please. That's all. Don't just say 1 plus 1 is 5. Those silly errors are anyways mathematical errors. Okay, we can practice these 5 questions more. I have bunch of more questions to practice on. Once you move forward, we'll do them more. Okay.
Can we take it as a homework?
Okay.
Yeah.
I can give you some.
Okay.
All right. Any questions, any issues, any concerns, any problems?